Good evening, everybody. Thank you for coming to this event that Vivo integration in Vivo in Vitro in study, and many also is the class basis research project presentation. And we have 4 groups. The first group is we call it CURE. They will tell us what CURE. And the first group presentation is Brendan. The slide will be set up by the IT group, and this is a video recording. Everything. Go ahead.
Hi, everybody. My name is Blake Mondrino, and today I'm joined by my partner,
Brandon, and we're here to present bacillus. Are you serious right now?
So as the global health crisis continues, scientists are researching to find new and effective antibiotics. Where are they going to find these antibiotics, you might ask? Well, you can find them in your backyard in the soil. Our task at hand was to identify, isolate, and run a series of tests on a certain bacterium to not only find their antibiotic resistance, but to also figure out what makes it thrive through a series of metabolic tests.
In the midst of those metabolic tests, we also took samples of our selected bacterial colony, BABM27, and ran it through PCR and NCBI nucleotide blast, all of which supported the notion that our strand was a strand of Bacillus cereus. So in order to obtain BABM27, we went through a large course of purifying and isolating various colonies. We did this by the spread plate technique, as shown in Figure 5 and Figure 6, where we diluted our soil within water, and we proceeded to spread them on these spread plates as shown here. We selected 32 colonies, 32 satellite colonies from a dilution plate labeled as 10 to the minus 3, and we spread plated them in order to analyze them later for zones of inhibition.
So we did a further zone of inhibition analysis, and with a plate that was impregnated with Serratia marcescens and Bacillus subtilis, we took the 3 bacteria that did produce cells of inhibition on our patch plates. That would be BABM4, 20, and 27. So when placed against Bacillus subtilis, neither of our 3 bacterium produced a zone of inhibition. But when placed against Serratia marcescens, the BABM27 did produce a zone of inhibition, so we chose that bacteria to be the subject of our further tests. So then from there, we did a streak plate to do further purification of our bacterium. However, this plate was not treated with cyclohexamide, which had been used in the previous plates to inhibit mold growth. Our plate did have quite a bit of mold growth. And because we're using this for the metabolic test and the PCR test, we do believe that this led to some of our error in the future.
I'm going to give a complete rundown over all the examinations that we made. These are some of the metabolic tests that we did. So the nitrate test came out negative, meaning that our bacteria does not have nitrogen reductase. Our thioglycolate test came back positive, as shown by the growth near the upper end of the tube, indicating that our bacteria is a aerobic bacteria. Our lactose dextrose fermentation test came back negative, meaning that our bacteria is unable to ferment lactose or dextrose, which was also confirmed by our litmus drug test, also coming back negative, showing that our bacteria is unable to ferment lactose and glucose. Our indole test also came back negative. Our urea test, interestingly enough, came back positive, showing that our bacteria has the urease enzyme, also demonstrating an area of potential research because of how we believe that we're unsure if our bacteria uses urease in order as a source of nitrogen, or if it just has it for other unknown reasons. And our oxidase test came back negative, meaning that our bacteria does not have cytochrome C oxidase, which is also quite interesting considering how all bacteria containing the cytochrome C oxidase enzyme, they are all aerobic, but not all aerobic bacteria contain this enzyme. And our MRVP test is where we believe there are some errors because of how our MR test came back positive and our VP test came back negative, when in reality, within our research, we found that Bacillus cereus should obtain a negative result for our methyl red test and a positive result for our VP test, indicating that our bacteria should have produced elements such as 2,3-butane diol and ethanol instead of mixed acids.
So the next metabolic test we ran were on the X plate. So the first core of the X plate was the Mueller-Hinton agar, which we used for the catalase test. This came back positive, meaning that our bacteria does have catalase, so it can convert hydrogen peroxide to oxygen. This is good because hydrogen peroxide is toxic to aerobic bacteria. For EMB, it came back negative, showing that our bacteria is not gram-negative, and it also does not ferment lactose, which is seen in the other metabolic tests. The next was KIA, once again, negative, it does not produce iron sulfide. And the citrate test came back positive, meaning the plate switched from a green to a blue. This means that our bacteria can use citrate as a carbon source. The final metabolic test we ran was on the starch plate, which came back as a positive, albeit a weak positive. But what this means is our bacteria can hydrolyze starch. It has the amylase enzyme. So what's interesting about this is only certain strains of Bacillus cereus have the amylase enzyme. If it doesn't have this enzyme, it means that it has the emetic toxin, which would lead to emesis. This is what you see in the strains of Bacillus cereus that are common in foodborne illnesses. In the midst of all of our metabolic tests, we sent it in for PCR analysis. When it came back, we ran it through the NCBI nucleotide blast, which furthered the notion that our bacteria is Bacillus cereus, which is a gram-positive aerobic or facultatively anaerobic motile organism, which is seen to be linked in non-gastrointestinal infections and is particularly devastating to the immunocompromised community. When we did the Sanger sequencing, the chromatogram came back. There were some blobs. There's very few repeating end sequences, some pretty sharp peaks. However, there was a lot of interference at the bottom, meaning that this also leads us to believe that there was a potential contamination.
Yeah, so within, although we had these examinations that yielded quite troubling results, such as what Blythe mentioned, the Sanger sequencing, some of the background noise that exists behind the overall peaks, and also within our MRVP test. All the rest of our metabolic tests, as well as PCR and nucleotide blast, supported the notion of our identification of VABM27 as a strain of Bacillus cereus. So throughout this course and this journey of various ups and downs, I get to say this one last time, which is why we named the project in this manner of Bacillus. Are you serious right now?
Does anybody have any questions?
We don't ask questions until the all group done.
Oh, okay.
So we will be coming back.
All right, sounds good.
This is the time pressure, so I let first group started. However, I skipped Dr. Zhou's introductory speeches. Dr. Zhou.
I have several pages prepared. Yeah, sorry about that. I had to step out for phone call, and they always come at the wrong time. First, welcome to today's event. During the next 2 hours, we'll have a number of presentations from different classes. That incorporated the inquiry-based research into the curriculum. So it is a quite well-anticipated event. I remember Dr. Chang talked about research-based curriculum when she was the chair of our department. Then during the academic year, I think 1890, and yeah, so she spearheaded the launch of the omics course-based research laboratory experience initiative with the support from the Dean's office for the purchase of yes, a lot of money from QIAGEN for the Ingenuity pathway analysis by biometric software, and for vision to integrate in Vivo, in Vitro, and in Silico studies in molecular and biomedical sciences. So when we talk about bioinformatics nowadays, we often think of the analysis of DNA and RNA data. even though bioinformatics analysis of the proteins is a critical component. Therefore, it is very easy for students and researchers alike to think that modern bioinformatics only emerged recently to assist the next generation sequencing data analysis. However, the very beginning of bioinformatics actually occurred more than 50 years ago. And when is that? More than 50 years ago?
Yeah,
Bioinformatics actually started in the 1960s. At that time, DNA could not be sequenced yet. However, the technologies were available to sequence what kind of macromolecule? Besides DNA, what's the other thing? Protein. Technologies were available to sequence proteins at the time. So computational methods were actually developed for the assembly of protein sequences and protein sequence databases. It was not until the late 1970s when DNA sequencing was available. What's the name of that DNA sequencing method? Sanger, that's right, exactly, Sanger sequencing. So during 1980s and 1990s, both biological science and computer science experienced a significant advancement. So the advancements in biological sciences allowed easier manipulation and the sequencing of DNA, while the advancements in computer science saw the rise of more miniaturized, it was not like the computer taking up the whole room, and more powerful computers, as well as novel software better suited for the bioinformatics analysis. So in the 1990s through the 2000s, there were major improvements in the sequencing technology, thanks to the human genome sequencing project. So these improvements led to reduced sequencing costs. It used to cost about, depends on how you calculate it, you can say 50 cents or 90 cents per base pair in our genome. How big is our genome? 3 billion. Yes, let's go for the upper end. 90 cents per base. How much would it cost? How much did it cost? Yeah, $2.7 billion. So that's at that time. And now we have something called $1,000 genome. That's a huge difference. So with the successful sequencing of many genomes and RNA samples, there comes an exponential growth or explosion of sequencing data. So that's what we call the big data. So universities and colleges have been coming up with various disciplines, such as the new terms, synthetic biology, systems biology, or wholesale modeling. So thankfully, Dr. Chang and the Dean's office were able to find a way to buy the Ingenuity pathway analysis from QIAGEN, which is a very powerful tool to analyze the bioinformatics data generated by our own laboratories or the data stored in public databases. So with this tool, we can use a three-pronged approach, that is in vivo, in vitro, and in silico studies, which will feed and promote each other. Today, we also see significant advances in both biological sciences and computer science. For example, have you heard of or used something called ChatGPT? GPT-4? Quantum computing? Artificial intelligence? Yeah? Yeah, definitely. So, but for biological science, many areas are also advancing very fast. For example, even for just the sequencing, the technologies are building upon the next generation sequencing with a higher throughput, more scalability, and faster speed. The new development will have, in combination with all those buzz things that you heard earlier, will have far-reaching impact on our understanding of life and biological research. So since the launch of the Omics Course-Based Research Laboratory Experience Initiative, Dr. Oso and Dr. Hill also implemented course-based research experience in the courses they teach. Dr. Oso has been infusing research into nursing, microbiology, and recombinant DNA courses. And Dr. Hill has been infusing research into his biology major, microbiology courses. So, sorry for the interruption of the presentation. But I would like to thank Dr. Chang, Dr. Hill, Dr. Oso, and you participants and guests. Without you, today's event would not be possible. And thank you to Dr. Chang for hosting today's event. So sorry for the interruption. Let's go back to the presentation. All right.
Insightful synopsis. Okay, so Holly?
No, that's us too. I think yeah, we'll work. Carly and Colette.
You are not sitting. That's me. Oh, I'm so sorry. I'm so sorry. Yeah.
Yeah. The second one from the bottom, the tiny. Yeah. Hi everybody, my name is Colette Catrona. This is my partner. So for our project, we decided to study in our microbiology class. We examined soil, and in this project, the purpose was to examine in the soil what bacteria in particular, if any, are capable of producing antibiotics. And this research is especially important today because in a world where so many bacteria are becoming resistant to so many antibiotics, this is crucial information that we can learn how some bacteria that we can find in our soil in very accessible environments can produce antibiotics that can be very useful in healthcare and a variety of other settings. So for this project, as I mentioned, we examined a sample from our soil, and through a variety of isolation techniques, we were able to identify a particular species and if it was capable of producing antibiotics.
So basically the purpose of our experiment was to discover any microorganisms that might have been living in the soil of surrounding areas. So the main purpose of this was to find out whether they had any antibiotic properties so that they can be used to possibly create new treatments for different diseases and disorders and to kind of alleviate the antibiotic resistance that's been prevalent. So basically when we took our microorganism, we did various tests. We put it on a patch plate, we discovered which one was presenting antibiotic properties, and then we took that one and we did a couple of fermentation tests. We did MRVP, catalase production, nitrate production, starch hydrolysis, urea hydrolysis, and a couple other tests to kind of see how it reacted and what kind of properties it had. So throughout all of our experiments, 6 of them turned out to be positive.
So just a bit about how we went about collecting our bacterial sample from our soil. I collected this sample from my backyard in Wayne, so a little different from the South Orange community. And on this day in particular, it was 83 out, not like it is today. And it was sunny out, there was minimal clouds and wind, so it was pretty easy to obtain the sample. You can see this image here. I tried to dig about like an inch or 2 into the soil to obtain my sample. And I wanted to pick an area that was relatively contaminant free, so I wanted to try and choose somewhere that wasn't super wet, there wasn't too many plants or rocks. I really wanted to see what exactly was in my soil. So you can see here, this is the tube that I collected it from. And then in class, we isolated using a patch plate technique. We created a patch plate here. And after letting it grow, we chose a particular sample that we saw a visible zone of inhibition. So for example, number 20, you can kind of see that there's a small zone of inhibition around that particular bacteria. And then to isolate single colonies using the PCR testing, we used the T-streak method, which you can see here, and we were able to successfully get a few single colonies, which we then ran in the testing.
So basically, this is just a list of the tests we performed and their results. So in the top graph, you have the tests that were performed in our test tubes. And out of those tests, 6 of them were positive. And then the second graph is for our X plate. So with the X plate, we had citrate, EMB, Clingers iron agar, and the Mueller agar. So let's get into that in a second. But in the bottom, we have the results of our PCR sequencing, which basically helped identify what bacteria we were working with.
So from our results, it was pretty clear to us based on using a dichotomous key that it was a Pseudomonas species, but the PCR testing helped us narrow down to in particular what strain of Pseudomonas it would be. As you can see, it was pretty clear that it was Pseudomonas. We've got a bunch of different strains, but as I mentioned, it was pretty clear from the results that it was a Pseudomonas species, but using that testing helped us narrow that down. So as we analyze the individual results, they're all pictured here. So these are our test tube tests, the metabolic tests that we conducted. The results for dextrose fermentation, which is this first one right here, were negative and negative. So that indicates that no acid or gas was produced and fermentation did not occur for our particular organism. The lactose tube is here, which proved our results of the dextrose test. Again, that no acid or gas was produced and fermentation did not occur. The nitrate test, which is in this tube here, was positive, indicating that the reductase ETS was present. The urea hydrolysis test was negative, meaning that this bacteria did not produce urease or ammonia, again, going along with the results of our other tests. The MR test was negative, showing that there was no mixed acid fermentation, and the VP test was also negative, so 2,3-butanediol was not produced. The litmus milk test determined the solution was alkaline based on fermentation and lysis products generated via the reaction. So across the board, it was pretty clear that no fermentation was occurring with our particular organism. Figure 2 in the top right here was our starch hydrolysis test, which indicates that the bacteria contains the amylase enzyme because it was capable of hydrolyzing starch. And then, as we mentioned in the previous slide, we also did some X plate tests. The citrate test came back positive, indicating that the bacteria is able to digest citrate. The EMB test determined that the bacteria were gram-negative, as the bacteria did in fact grow on that section of the plate. So that was also a very large indicator that our bacteria was likely a Pseudomonas species. And the Clingers iron agar also demonstrated a negative result.
So through our metabolic test, we were able to use a dichotomous key to kind of identify what bacteria we were working with. So using the test and that key, we narrowed it down to a Pseudomonas species. Once we got our PCR results, we were able to confirm that it was a Pseudomonas species, but it specifically pointed to the QDF12 strain. So part of why we were able to identify that it was a Pseudomonas species was because of the gram-negative test on the EMB plate and then the negative lactose fermentation test.
And all of our tests that we conducted were pretty much in agreement, so there wasn't really anything, as we were just using the dichotomous key to prove that was a Pseudomonas species. And since all of the results were kind of in unanimity, it was pretty easy to make that determination, and the PCR testing confirmed which strain in particular this was. So based off of our observations and from this determination, we can answer the research question that asks us to analyze the antibiotic properties found in bacteria that can be in common soil nearby. So this proves that even, anywhere, somewhere so local as your backyard, in my case, did in fact have the capability to produce antibiotics, as we observed on the patch plate and as we observed clearly based on our testing. So in particular, Pseudomonas species are pretty common and found in soil and water environments, and some particular antibiotics that they are known to produce include phenazine, quinoline, and pyrrole derivatives, and heterocyclic quinones. These potential antibiotics produced can explain the zone of inhibition that we observed on the patch plate, and therefore, based on the results, we can conclude that our bacteria number 20 from the patch plate was likely an antibiotic-producing Pseudomonas species. Thank you.
like that.
Oh my gosh, I'm so sorry.
Thank
Please. Oh man, but it's good news. Okay.
All right, so thank you. Okay, so good afternoon, everyone. My name is Natalie Irwin, and today I will be presenting the research that my partner and I did for the Tiny Earth project, where we isolated and identified a bacterium that we named AKNI12. And so this project presented us with a chance to contribute to novel antibiotic research. And this information is critical as the antibiotic resistance crisis continues to compromise the efficacy of fundamental antibiotics on resistant pathogens. So we felt proud to gather information that could be applied to the development of future antibiotics. So in the beginning of the 10 weeks, we collected a sample from the South Mountain Reservation. It was about a late fall day, so it was about 70 degrees in the air. And we were using, so we used a conical, and we brought this conical into the lab, where we then hypothesized that this bacteria will probably present antibiotics that are mildly to extremely resistant because of the environment it grew up in, because it was on a riverbed. So we figured that it had to face much competition with the other microbes in the water and in the soil. So in the next steps, we diluted our sample and poured it onto a spread plate of LB or Luria broth agar. So we allowed the plate to develop for about over a week, and we used an aseptic technique where we selected 32 colonies and placed them onto another more organized patch plate. And from the patch plate, we're better able to identify the strongest antibiotic producers. So in the end, we chose AKNI12 because of its ability to resist the other microbes on the patch plate. So we figured, what can it do with the other pathogenic microbes, as well as the other gram-negative and gram-positive microbes that we have in the lab. So we observed AKNI's ability to produce antibiotics against Erwinia, E. raffinensis, and Acinetobacter, and found it inhibited the growth of all three. So then we determined AKNI12's metabolic abilities. From the series of tests, we declared the AKNI12 to be a facultative anaerobe, and this had the ability to ferment dextrose, as well as use citrate as a carbon source. So additionally, we confirmed the AKNI12's ability to produce catalase, amylase, and cytochrome oxidase. So these characteristics gave us the hint as to what species of bacteria we were working with. So using an outsourced company, Dr. Hill sent our organism out to another company where they sequenced our DNA for us. And we were able to put the amplified DNA code into the BLAST sequencing database. And we identified the species of AKNI12 to be a type of Pseudomonas. The DNA of our species had a 90% match with Pseudomonas mucili, as well as a 90% match with another Pseudomonas called Pseudomonas fluorescens. So this meant there's a high probability it could be one of the two. And so this, knowing this, we were a little bit confused, a little concerned, but we were able to refer to the dichotomous keys of Bergey's manual. And this manual was a great manual to use for assistance because we were able to take the metabolic results we got and apply them and narrow it down to what type of species, what type of Pseudomonas AKNI12 could be. So unfortunately, our metabolic results did not exactly match the results of their species, being Sulie and fluorescens. So we worry this may have been caused by an error in our technique. So the contradictory results were puzzling. So we discussed the possible limitations that we faced and the sources of error that we produced during all of this, some of which included difficulty isolating bacteria in the first place, genetic mutation through horizontal gene transfer, and then faulty aseptic technique, given that this is our first time in a microbiology lab, and perhaps contamination within the growth media. So in the future, it would be beneficial to repeat the experiment, but use a T-streaking method instead. So this way we could, so using the newly isolated Pseudomonas colonies, we could run additional metabolic tests that are specific to the Pseudomonas genus. And this would include testing for fluorescence, arginine, trehalose, denitrification, as well as the metabolism of gelatin. So with these results, we can refer to the more advanced hypothesis keys of Bergey's manual that would allude to the Pseudomonas genus. So once the species is confirmed, we would continue to test the exceptional ability exceptional ability of this AKNI12 to produce antibiotics. And furthermore, the antibiotic compound could be extracted and analyzed to understand the biochemical mechanisms it uses to fight resistant bacteria. In sum, this information will provide further insight into how to eradicate the current antibiotic resistance crisis. Thank you.
The next presentation of this group is Diana.
So hi everyone, my name is Diana Malakova, and today I would like to present some research that we did on bacteria, antibiotic-producing bacteria in soil. So all of you have the abstract, so I'll probably get going. So introduction. Why is this research important? So antibiotic resistance is a global problem that has been going on and is just progressing. People misuse antibiotics, people don't finish antibiotics to term. In other cases, bacteria gets more and more resistant as animals get fed antibiotics, as people misuse antibiotics. So in all, bacteria gets more resistant, and this causes pathogens, diseases to get more stronger. So with the Tiny Earth project that was designed, we're trying to figure out what other antibiotics we can find, if there are any new subtypes of antibiotics, and if some antibiotics can be stronger than the counterparts that we have right now. So first we'll start with the soil extraction. So the soil that I tried to choose something that is mutated. So I went to Bridgewater, New Jersey, that's where I live. And the reason why I chose the soil is because it's really hard and clay-like. So there's limited growth or plant growth there. As well, you can see that there are plants growing there, but it's not abundant. It has a ton of sunlight, so I was thinking of UV radiation. And finally, it's located right below an artesian water tap. And I thought this was important because artesian water is from the underground wells, and this means that it's less altered than water that we would get from like the community sewers. So also this area is historically industrialized, so it has a bunch of trains, lots of air pollution. So I was thinking this bacteria could be a little bit mutated than the rest that we can find. So methods. So first of all, as I explained, we collected soil samples. Then we had to dilute samples into, to kind of narrow down the amount of bacteria that we were finding in the end. So from the collection that we had, we narrowed it down to like a library, and we played it until a place for the library to use. Finally, after the dilution and isolation of a library, we used this library and plated it on E. coli and Staph epi. This gave us an opportunity to observe halos, or if the halos are present. So as you can see, there is sample number 13 on the top and middle, and the halos indicate that the bacteria fights against E. coli or Staph epi, which is good. Means it produces antibiotics. So after that, we used the sample number 13. There was actually two samples. It was sample 13, sample 8. And we plated them onto this plate below in order to look if it fights against other bacteria such as B. subtilis, E. raffi, other examples. And the halos that were more prominent were for bacteria number 13. So that was the bacteria that we used to progress on. So, there was also, after that, we used metabolic testing and PCR, but I'll get to that. So here are the metabolic tests that we used. So as you can see, we had positive for dextrose, for lactose, for urea hydrolysis, starch hydrolysis, catalase production, hydrogen sulfide, and citrate fermentation. And we had negatives for our MRBP test, nitrate production, indole, and oxidase. So overall, from the X plate that you can see below, we figured out that the bacteria is a gram-negative, and it's also a it's a fermenter, so it can ferment dextrose and lactose. And later on, we figure out that it's facultative aerobe because from the test that we did, it could be present both on the top and on the bottom of the test tube. So using the metabolic testing, we decided to use a flow chart or a dichotomous key in order to see what bacteria it could possibly be. Once again, this, as you can see, this doesn't have all types of bacteria, so it's not as accurate. So we kind of took this with a grain of salt. But based on our test, we looked at what bacteria can be, and it turned out to be Citrobacter fermenting. In order to verify that, we did PCR, or Dr. Hill did PCR for us. As you can see, there was limited interference, which means that our sample was not contaminated. It was pretty neat. And based on that, we ran a blast-in procedure used by the NIH. And with that, it gave us that there's a 97 to 98% similarity with Pseudomonas genus, which means that it is in some shape or form, a variation of Pseudomonas. This obviously didn't match with our metabolic testing dichotomous key, so here we had to see which one we would trust more. We decided to make a group, my partner and I, we decided to make a group and to compare the unknown to Pseudomonas. And one major difference was that the oxidase was negative for us, but it's always positive, or usually positive for Pseudomonas bacteria. And another difference was the hydrogen sulfide production. But as you can see from the graph, it can be variable from Pseudomonas to Pseudomonas types, so it's never the same. So the question comes, what do we trust? So PCR results were a little bit more specific, and there is 97 to 98% chance that there's some Pseudomonas. So we decided to go with the PCR results rather than metabolic tests. Once again, metabolic test, the dichotomous key, did not have as much bacteria variations, so obviously it could not account for everything. And once again, because of the mutations, it is possible that the Pseudomonas could differ a little bit metabolically, but the nucleotide sequencing still showed 97% similarity. So once again, there's reasonable similarity between Pseudomonas and PCR, and the verdict is probably a variation of Pseudomonas. So conclusions, or what did we learn from this project? We learned that our bacteria is a gram-negative facultative anaerobe, and that it's likely Pseudomonas genus. Pseudomonas is, once again, a gram-negative, and it is kind of a really nasty bacteria when it comes to hospitals. So it can cause pneumonia, it can cause blood irregulations, and this is more prominent in hospitals than in regular life, so it's not really harmful when you interact with it in soil samples, for example. And once again, Pseudomonas can provide a wide variety of antibiotics, such as heterocyclic antibiotics. There's an example of phenazine, which is commonly used. And from testing, we figure out that both PCR and metabolic tests can be used to look at soil samples and bacteria. However, PCR can be trusted sometimes a little bit more because of the sequencing, and that it's a little bit more accurate because it looks at the nucleotide sequencing. Possible errors. Obviously, no experiment goes without errors, and we were no exception. Metabolic tests can be similar from an organism to an organism, so it would be hard to tell them apart. So for some reason, if you have some variations of Pseudomonas, they might have a similar metabolic test, or they might have different metabolic tests, but the dichotomous key can identify them as two different organisms. And once again, our observations are based on perception. So for example, if I go back, there was a starch plate, which appeared blue. For us, we did see halos, so we think that the starch catalase was positive. Starch hydrolysis was positive. But for some people, it might appear more negative. So once again, observations are based on perception, so that's the human flaw that goes into it. And PCR testing can also have interference. We had limited interference, but some of my colleagues they had a lot more interference, and that altered the results. So it's like hit or miss. So importance, new antibiotic discovery. So soil health profile, and this is important because we can see what bacteria are present in the soil, what fights against what. Is the soil good for example, food planting? Can we use it for food? And once again, we can look at interactions between bacteria. This always gives insight into what organisms we can use against each other, or just in general, it can give us insight about our world. Future directions. So what we didn't do in the lab, we did not extract the antibiotic. We did not do the antibiotic testing on animal species, and obviously there was no clinical trials. But perhaps this gives us future purpose to look at the antibiotics, see if this antibiotic exists, and perhaps test it if it can be used for clinical purposes. And that's it.
a question. Diana, you may want to stay.
Oh, stay?
Yeah. One question. And come to the front.
Okay.
Almost? Yes.
Yeah, I think those four groups, when I go up to
We
the.
have about 5 to 6 minutes for question for the group. You can ask each other or the audience for a question. Yes.
I have a question for Dan.
Can you please tell your name?
Yes, my name is Hannah. I have a question. You mentioned that Pseudomonas is particularly harmful for those in hospitals as opposed to just interacting with it in the soil. Is that just because of the patients? I guess if you're in the hospital, you're already maybe immunocompromised, or are there different levels in hospitals?
So from what I read, yes, it's because of the patients being immunocompromised, but at the same time, hospitals also use a lot more sterilization techniques, and so it might be possible that the Pseudomonas present there is a little bit more harmful or more mutated to be more resistant against bacteria. But just in general, from what I read, yes, it's about like immunocompromised patients. Thank you.
Did you guys find any new antibiotics?
Well, I can answer. Where are you gonna go?
You're on a roll.
So we were not able to look at, we don't know what the antibiotics were. So as we said, we found that they exist when bacteria produces them, but we did not get to actually synthesize or see chemically what antibiotic it is to see if it's present or not.
Going off of that, I think we can all agree that if we're going to do further trials, we would look further into what antibiotics were produced by our bacteria to try to further what we learned in projects.
You could just do PCR and find genes or coding for the different antibiotics in there, possibly, and then place it into a yeast or something.
Hi, my name is Tatiana. I'm a graduate student, and this is actually a question for the first group, Brendan and Blythe. I'm sorry if I mispronounced that.
No, you pronounced it perfect.
So you guys said that one of your tests, unfortunately, I don't remember which one, I'm so sorry. There was like a contradictory kind of result compared to what you were expecting. Could you go into maybe a little bit of detail how that could have happened?
Yeah, of course. So basically, that was our MRVP test, which stands for methyl red bogus Proskauer test. They both use the same solution, and basically in the examination. So for methyl red, right, you use a substance called methyl red, and if your bacteria produces mixed acids as a result of like fermentation products from sugar and things like that, then the acidity of the solution will increase and the methyl red test will appear red because of the decrease in pH. And for the bogus Proskauer test, a similar effect, a similar like visual effect is displayed, but instead of in response to like mixed acids, it's in response to like 2,3-butanediol and ethanol. So our results when compared to things we saw online, things we researched of Bacillus cereus, they were completely opposite. So for us, our sample yielded a positive result for methyl red, when it's negative for the most part for other strains of Bacillus cereus. And then we yielded a negative test for VP, bogus Proskauer test. When obviously when compared to other strains of Bacillus cereus, this is usually positive. So
when we did the dilution streaking, that plate that we used, previously all the plates that we're using have been treated with cyclohexamide, which Dr. Hill told us, but that plate that we did the T-streaking on wasn't, and we got that plate back the next week. There were 2 like gigantic spots of mold on it.
yes.
So there was some contamination in our sample, and we believe that that is what gave that conflicting result. You could also see it in the Sanger sequencing. There was a lot of background noise on it, but all the other metabolic tests were in line with Bacillus cereus. So, and because of the nucleotide blast, we went with that as what we decided our bacteria was most closest to.
Yeah, and just one last thing, within our test, you could kind of see it's a bit small to see on screen, but there was mold growth within the top of both test tubes. So.
Okay, thank you so much.
No problem.
Yeah, hi.
My name is Wangfei Huang. Thank you for your nice presentations. So my question is, my question is actually basically maybe for every presenter. So I know like there are like various like kind of species of bacteria in your soil samples. So why you all study like one of them, like kind of how do you select those one of the bacteria you select to study?
I can speak on that. So as much as I would love to probably, analyze every possible bacteria that could have been in that soil, from ours, it was pretty clear as we did the patch plate, which one kind of had a visible zone of inhibition around that particular bacterial species sample in that particular colony. So we chose it based off of visually which one we thought was going to produce antibiotics. So if there was no visual zone of inhibition around a specific bacteria sample, then we didn't choose that one because likely, maybe possibly, who knows, if we tested all of them, it could have. But based off of the time frame and based off of the media that was on, we chose our bacteria sample to study based off of that visual appearance, if there was a zone of inhibition or not. And that would indicate if it was likely producing antibiotics, which in our case it probably was.
Any more questions? Okay. Thank you so much for the next group is Dr. Luce's class, which has two group projects. And the first one is on law of O2 semantic donation and the system 239 in membrane trafficking. Paul Young is going to be the presenter.
Hi, I'm Paul. I'll be presenting research conducted by myself and my peers in Dr. Urso's class for recombinant DNA technology. And I'll be on the role of GLUT2 as palmitoylation at cysteine 239 in membrane trafficking. So palmitoylation is a reversible post-translational translational modification where palmitic acid is added to a protein. And this changes the protein's hydrophobicity in turn, as well as the membrane affinity. And this could affect the various features of the cellular mechanisms. And as of recently, glucose transporters 1 and 4 have been found to be palmitoylated. And they were found to influence different kinds of things, such as promoting glioblastoma glycolysis in GLUT1 and movement in response to insulin for GLUT4. And both of these glucose transporters belong to the class 1 isoform, in which they share similar structures as well as conserved residues or conserved sequences. But it's still unknown if any other isoforms are found to be regulated through this modification. So GLUT2 is the major glucose transporter of the liver. It plays roles in maintaining glucose homeostasis in feeding and fasting states. It has relatively high affinity for glucose and fructose, and it is also seen to mediate glucose signaling pathways from the membrane to the nucleus. So our basic schematic is we did an initial acyl PEG assay, which is an assay that pretty much tells us if it's palmitoylated or not, and not directly, but it could give us some sort of idea. And we also performed a bioinformatics analysis to determine which cysteine residue will be of interest. And we also stably transfected cell lines, wild type, containing no mutation, as well as a mutant in which the cysteine residue was mutated to a serine residue. So therefore, no palmitylation should be occurring in those cell lines. We performed the AP assay, confocal microscopy, MTT, and the PCR assays. So for the results, this is the initial Western blot analysis of the MPEG APE analysis, not the APE analysis, but the mass transfers analysis that we did with the GLUT2 antibody. So essentially, the hydroxylamine takes off palmitoylated groups and gets replaced by this mass, which causes for the shift, for the upward shift becoming, because the mass tag is heavier, about 10 kilodaltons. So on the left here, this shows that with the treatment of the hydroxylamine and the shift in the band here with the no presence of the band in this lane tells us that there might be a palmitoylated group within GLUT2. And this is right around the size of a minor form of GLUT2, which is non-glycosylated. So our bioinformatic analysis, we used a program called GPS Palm, which conveniently tells us which cysteine residue will be most likely palmitoylated. And cysteine 239 showed the highest score with 0.835 as shown here. So based off of this, we also use Unipron program to determine the to obtain the amino acid sequences as well as the transmembrane domains. And based off of that, we were able to construct the schematic structure diagram of GLUT2 and place the relative cysteine residues that the GPS POM program gave us onto their sites. And cysteine through 239 was present between transmembrane 6 and 7, and conveniently it was within the cytosol, and as well as around the same area as the other known cysteine residues for GLUT1 and 4. So the acyl PEG exchange assay, or the AP assay. So in the top box here, before I get into this assay, I just want to let you guys know there are two forms of GLUT2 proteins. One is non-glycosylated and one is glycosylated. The glycosylated form is heavier, and non-glycosylated form is a little bit lighter, but it's the minor form, in which the bands might be showing a little lighter, but it's the same, essentially it's the same, we're looking for the same thing. In the wild type, we should see a mass shift of about 10 kilodaltons per palmitoylated group. And in the fourth lane with the 2-BRPA, that's the inhibitor of palmitylation. So we shouldn't see intensity in bands where there was intensity within the wild type. We should see around the same thing for the mutant as well, because palmitylation should not be occurring there. So it might be a little light, but in the whole cell lysate, we see a band around where the non-glycosylated GLUT2 protein should be, and we see we see the upward increase around of around 92, which is around one palmitoylated group, and we see and we also see a clearance compared to the other bands here, albeit it might be not as clear because there's some background signaling. It is more clear in the more major form of the GLUT2 protein, with the shift from around 99 to 119, which is around 20 kilodaltons, which could mean that there could be multiple cysteine residues that are palmitoylated within this protein. And then you can see that the band over here, it's a little lighter than the inhibitor as well as the mutant. This is our confocal microscopy assay. So we see that in the wild type GLUT2, in the top three rows, the GLUT2 protein is more localized, but generally more localized around the membrane, whereas in the mutant, it's present more inside the cell in vesicle-like formations, albeit there are some still that reside in the membrane as well. So we also performed an MTT assay within complete media to see their proliferation within 24, 48, and 72 and 96 hour increments. So interestingly enough, the mutant cell line showed a higher proliferation compared to the wild type and the untransfected line. So we tried different sugar states to see if that would change anything. And the results were consistent within the hyperglycemic state. The mutants showed higher viability compared to the other cell lines. The explanation for this is still unclear. There's different hypotheses that we do have, but it could be due to that different states of palmitylation could traffic the membrane to different lipid kind of microdomains, whether that be in the raft or generally around the membrane. Different kinds of phospholipids present in those microenvironments could alter their function to maybe intake glucose or even perhaps send signals down to the nucleus from the plasma membrane as well. So more relating to that, we also performed PCR just to see if they changed gene expression on a FASN and PECK1 genes. And consistent with our previous MTT assay results, the mutants seem to increase the expression of these respective genes. So there is a clear, distinct difference between the wild type and the untransfected compared to the mutant, but we're not exactly sure how the palmitylation is inducing these kinds of changes yet. So the next step will be potentially to, because these mutant and the wild type cell lines are transfected, they're also still expressing the endogenous GLUT2 proteins, which means the proteins that are produced by the cells themselves without the transfection. So inhibiting the production of these endogenous protein with maybe RNA interference could give us more clarity in terms of what the fusion proteins are doing. And we could also perform more PCR on various downstream targets of glucose signaling pathway as well to determine how they affect gene expression. And another thing is perhaps insulin signaling pathway could also be looked at because GLUT4 moves internalizes within palmitylation, and GLUT4 is related to insulin signaling in that way. And GLUT2 is also recruited to the lipid raft to become internalized with insulin receptor as well. So that maybe palmitilation has a role to do with that. And I'd like to thank Dr. Urso again and Dr. Chang for hosting this event. And those
Done by Hannah. The title is Combination of Group 3 at 16205 mediates Group 3 membrane localization and function.
Hi, everybody. Good evening. Thanks, Dr. Chang, again for hosting us this evening. My name is Hannah, and I'll be presenting on behalf of my co-contributors and teammates on the palmitilation of GLUT3 at cysteine 205, which we believe to have mediated the GLUT3 membrane localization and function. So really briefly, piggybacking off Paul, what is palmitilation? Why do we care about it? How does it work? So palmitilation is a reversible post-translational modification to proteins that is going to help anchor proteins to the plasma membrane, as we see in this image here, and can attach to a transmembrane protein, anchoring it in spot, affecting its availability in the plasma membrane. So before we began our experiment, it was previously already known and found that GLUT1 and GLUT4 are palmitilated at the plasma membrane region, affecting their availability in the membrane to transport glucose in response to glucose needs of the cell and external signals. So then we began to look at the class 1 GLUTs as well. So we looked at GLUT2 and GLUT3, and we tried to run bioinformatics to determine what we think would be the next best GLUT to test for palmitilation. Via uniprot protein analysis, we were able to sequence these genes, and we ran through the sequence of all the GLUTs class family glucose transporters and found a conserved region of the cysteines that were known to be palmitilated in GLUTs 1 and 4. And we found this region of the conserved sequence at cysteine 205 and GLUT3. And then we also ran interspecies protein alignments to show the evolutionary benefit of keeping this remaining conserved sequence, and we noticed that in GLUT3, this sequence via the interspecies alignment remained consistent, which hinted to us that this might be really important for its function. And then we also used the protein alignment to determine the transmembrane region and found that similar to GLUT1 and 4 that was known to be palmitilated, this cysteine of interest at 205 is again outside of the transmembrane region, available to be palmitilated. So the importance of GLUT3 as the neuronal transporter, it provides mostly neuronal cells with the glucose as needed for the metabolic needs of the cells. And via the use of GPS-PALM, we also determined the likelihood of palmitilation at this conserved cysteine site as a likelihood of 0.08005 on a scale of 0 to 1. So we believe that there was a high likelihood of palmitilation, and so we continued on. So for our methods, after we confirmed with bioinformatics that we were interested in this specific GLUT isoform, we transfected wild type, mutant cells that I'll show later, gave the cells various treatments to run various experiments and assays to determine if palmitilation is seen in some of these cells. So I included our figures. I think I can speak on behalf of our team that we love these figures. We love how they turned out. So really quickly, I'll kind of show you what's going on here. So this green staining is staining the plasma membrane screen of these neuronal cells. We also tagged the red end cherry to our transfected GLUTs. And then over here, you'll see also there's a blue that stained the nucleus blue, or the nuclei. So in figure 2 on the side over here, we have 2 treatment groups of DMSO and a palmitilation inhibitor. So in this first row, we see DMSO as our control. This is supposed to be our neuronal cells in their normal resting states. And we can see that in these resting states, there's a superimposed image of this red and green, or the glucose in the membrane. That's what we believed. And then when we put in the palmitilation inhibitor, the 2-bromopalmitate, we saw a shift of the GLUTs away from the membrane, which we believe to be inside the cytoplasm, indicating to us that, hey, maybe palmitilation is really important for the plasma membrane localization. So we continued on. And again, we have our stainings here, and we have our respective treatments of wild type GLUT, the GLUTs in their natural habitat,
And then we also transfected a mutant type, changing the cysteine of interest that's able to be polymethylated to a serine, reducing the GLUT3's ability to be polymethylated at that site. And we saw something similar. So in the wild type that's able to be polymethylated, we saw that, again, the superimposed green and red, GLUT3 in the plasma membrane. And then when we added the mutant type instead of the wild type, we saw that, whoa, a shift again. It's no longer able to be found in the plasma membrane. It's outside of the nucleus. We hypothesized that with the mutant, the GLUT3 was able to internalize away from the plasma membrane because it's no longer to be able to be polymethylated in between the plasma membrane and the nucleus, resting in the cytoplasm. Okay, so again, more data to kind of further our suspicions that it's polymethylated. Now we also ran an APE-ESA or acyl-PEG exchange, which basically means that we added this 5 kilodalton PEG tag, and it's going to add itself wherever there is polymethylation, if any. So really briefly, when we look at this, we can see that our GLUT3 wild type, which is subject to polymethylation, it's resting here at about 89 kilodalton. This is against the cells with the polymethylation inhibitor and the cysteine that was mutated to a serine. So these cells are no longer able to be polymethylated, and we see roughly a 5 kilodalton difference from 89 to 84 kilodaltons, again, supporting our hypothesis that GLUT3 is polymethylated. So we're really excited about that. So now that we've supported our hypothesis that GLUT3 is polymethylated at this site, we wanted to determine what does it regulate, why do we care? So we ran a cell viability assay or metabolic assay to determine basically how much glucose is being pulled into the cell, because as we all know, glucose is energy. So with the wild type cells that are able to be polymethylated, able to be in the plasma membrane, we're hypothesizing that this affects the wild type's ability to bring in glucose because it's in the membrane and it can reach extracellular and bring the glucose into the cytosol. So with those wild types that are in the membrane, we believe are polymethylated, we see a significant statistical increase in metabolic activity or glucose transport into the cell versus the neuronal N2A cells that are untransfected, as well as the cells with the mutated form, as Paul said, based on the endogenous pre-existing glutes in the membranes, but the transfected wild types were able to pull in more glucose. So now that we know that polymethylation aids the GLUT3's ability to be in the plasma membrane, and also encourages the GLUT to be able to pull in glucose as per the cell's needs if need be, we wanted to know, what does this mean downstream? So we also took a few genes that are related to metabolic activity, cell growth. Like I said, glucose is energy, so now we can grow the cells, we can start activities within the cell. And we ran PCR. And we saw that with the wild type glutes that have increased metabolic activity, increased glucose transportability, we saw this increase in genes that promote cell transcription activity as compared to our control beta-actin. And kind of interestingly enough, we saw IRS1, which is another gene that we thought would significantly increase in the wild type. It was slightly increased, but this minor increase versus these also indicated to us that GLUT3 has significant regulation only on certain genes. So we know that it does impact genes, but now we're interested in using IPA software, which is able to kind of give us a hint of certain genes. So I was able to link GLUT3 to the left here, neurodegenerative disease on the right here, and see a full list of genes that we're kind of sifting through right now, as well as hypothetically plugging in our data. So if we know that ELK1, the transcription activator, if it's upregulated, then we can inhibit neurodegenerative disease because it's keeping the cell alive. Same with IRAK3, if we upregulate this, which is neuroprotective, we're also going to decrease the susceptibility to disease. So our conclusions, like I said, we have supported the idea that polymethylation is occurring at GLUT3 cysteine 205, and that it regulates GLUT3's ability to be found in the plasma membrane, as well as regulating the cell's metabolic activity, as well as what we're interested in learning next is the development of certain diseases.
again, I have my team, sorry, don't clap yet. I'm here with my team today, Dr. Urso, so we're really excited to have been able to share this with you. Now, oh yeah, there's it.
It's regarding, my name is Mohammed Bashir, I'm a grad
Louder,
student. This is regarding the GLUT3. So what do you think in the central nervous system, the GLUT3 palmitoylation, whether it contributes to the insulin, how it contributes to the insulin resistance, or like, how it manages the glucose transport? Like, by keeping it in the plasma, by localizing it? Or
Oh, how it regulates glucose import?
Yeah.
Okay, so very generally, if the glucose transporter was not able to be in the membrane, it wouldn't be able to reach both the extracellular space, transport glucose from around the neurons to inside. So the palmitylation is able to use it to the membrane and keep it there so that it can keep bringing in glucose, versus if the palmitylation leaves, the protein might also leave the membrane and then not be able to import glucose. So if the cell happens to need glucose, they're down or upstream signals, I guess, that would signal palmitylation to come, and then anchor it to the membrane until there's no more glucose that needs to be in the cell. Anything else?
So mutations in the palmitylation of GLUT3 transporter can cause disease, correct? Or is there no indication of such mutations in medical literature, or have you done any research on that?
Yeah, so we also hypothesized, so it's kind of based on the glucose in the cell. So if we looked at the mutated form that's not able to be palmitylated, then the cell wouldn't be able to have that extra glucose coming from GLUT3. So then we're looking at the genes associated with neurodegenerative disease. If we have a lack of glucose, what happens to the cell? Like chronic lack of glucose, then there might be some disease. The cells might just die, right? Because they just don't have the energy. But there are also genes associated with just hyper and hypo glycemic levels as well.
But there are no studies done to a Vitro or Vivo study.
Yeah, we're really excited. We hope so.
I have a question. How did you make those mutants?
We did site-directed mutagenesis. So we constructed our respective forward and reverse primers. And essentially when we do the PCR with them, it copies everything correctly except for that one nucleotide that we changed within within the promoters that we constructed ourselves.
I have a question, Eric Hill. Why serine?
I can answer that. So with serine, it's the closest, you could say, like amino acid in terms of structure to cysteine. Another thing is that the S-palmitylation, it specifically has to deal with the thioester link that cysteine has. So we decided to take that serine because it's the closest, and it actually did work as a mutant form.
It did work, thankfully, yeah.
The other question I have is, how do you know there are these disulfide bridges that work? Was that positional serine involved in the disulfide bridge, or was it not?
You'd have to do the analysis of the protein itself for that. So that's pretty difficult. You X-ray just a lot of you know what you.
You can do DTT or be involved.
Can I follow up on your question? So that, sorry, if we don't do that, that could provide like confounding data, is what you're getting at?
Whether or not it's not making it to the surface, if it's misfolding, because it's a structural intact, anytime you start removing cysteines, you can remove some, but usually you have to remove pairs so that it still kind of remains. And if it's a bonding one, then it becomes more likely to be ubiquitinated or destroyed, or it's not going to make it to where it's going to be expressed. And if you add another palmitylation, it's just making more fatty acids. It's just going to start globbing on other membranes where it finds some other hydrophobic regions.
Thank you.
Any more questions? Okay, thank you for the wonderful presentation. Okay, the next presentation will be done by Shama Kavya. The title is the network meta-analysis of STAT3 involvement in ethanol augmentation of thymoma.
Hello, everyone. So the thymus is an important organ of the lymphatic system, and it serves as the site for T cell maturation. And there are two types of thymic cells. There are thymocytes that mature into T cells, and there are thymic epithelial cells, which form the outside layer of the thymus. Thymoma is a neoplasm of the thymus that affects thymus epithelial cells, or TECs. It is a part of a group of thymic cancers named thymic epithelial tumors, and it is often linked with autoimmune paraneoplastic diseases such as MG, HGGB, and PRCA. The majority of thymomas are encapsulated, but 31% of cases metastasize. The consumption of alcohol is a known human carcinogen. Its metabolite, acetaldehyde, damages DNA and cells' abilities to repair damages. Ethanol is the principal compound in an alcoholic beverage, and it leads to inflammation of the body through its activation of cytokines and the inflammational signaling pathways. Ethanol also leads to an increase in endogenous glucocorticoid levels, which have immunosuppressive effects. Ethanol has also shown to lead to thymic atrophy in a study of female mice, and it led to apoptosis of those thymocytes, but not the epithelial cells. So perhaps there's an opposite effect on those TECs. Signal transducer and activator of transcription 3, or STAT3, relays signals from cytokines and growth factor receptors in the cell membrane to the nucleus to regulate gene transcription. In the immune system, it signals for the maturation of immune cells, such as T cells. STAT3 expression is elevated by ethanol exposure, and through ethanol, STAT3 can increase cell proliferation, migration, and apoptosis. Elevation of STAT3 can therefore cause DNA damage and mutation, which can lead to that increased risk of cancer. So my guiding question was, can ethanol exposure lead to the progression of thymoma through the STAT3 pathway? So the method I used in the study was QIAGEN Ingenuity pathway analysis, or IPA, which is a bioinformatics software that allows for the exploration of nodes through created research. I used the GROW tool, which produces all the chemical molecules associated with the node, the Path Explorer tool, which connects those molecules together, the TRIM tool, which removes molecules that fit a desired criteria, the molecular activity predictor tool, which allows for the simulation of increase or decrease measurement, as well as IPA core analysis, which displays upstream analysis and canonical pathways. And using these tools and data collected from the QIAGEN knowledge base, I constructed a connectivity map. So my first connectivity map was between ethanol and STAT3. I used the GROW tool on the nodes of ethanol and STAT3, and using the Path Explorer tool, I found 226 intermediary molecules. 7 of these molecules were removed as they had an upstream effect on ethanol, and my study is related to the downstream effects of ethanol. And with the map tool, I increased ethanol measurement, and this led to an overall activation of STAT3. Using the TRIM tool, I removed 73 intermediary molecules as they had no measured effect on STAT3 expression, and this left me with 146 intermediary molecules. Then I performed core analysis on these intermediary molecules, and I found the top 10 canonical pathways involved, with hepatic fibrosis, neuroinflammation signaling pathway, and glucocorticoid receptors as the top 3 most significant canonical pathways. I also would like to note that molecular mechanisms of cancer is also included in this list, so keep an eye out for that. I also performed the upstream regulator analysis, which presented me with the top 10 upstream regulators. Basically, the upstream regulators are those intermediary molecules, and these are the top 10 which had the most profound effects on STAT3. TNF, IFNG, insulin, IL-6, and TGF-B1 are those top 5 upstream regulators, and it is important to note that the top 3 here, minus insulin, are cytokines, which hint towards that heightened inflammational response. Then I used GROW tool on STAT3 and thymoma, and I found, using PathExplorer tool, 18 overlapping molecules. 3 of these molecules were removed as they had that upstream effect on STAT3. And using the map tool, I increased STAT3 measurement, which led to that overall activation of thymoma. Using the trim tool, I removed 11 of these intermediary molecules because they had no measured effect on thymoma expression. So given that STAT3 expression leads to thymoma activation and an increased ethanol measurement leads to STAT3 expression, I can logically connect these two connectivity maps together and see the overall effect of this pathway. So I used PathExplorer to do just that, and an overlap occurred between ethanol and the intermediary molecules between STAT3 and thymoma. But since I want to look at a step-by-step map of the study, I removed those overlaps. So as depicted in previous results, an increased ethanol measurement led to the activation of STAT3 and subsequent progression of thymoma. I also performed core analysis on the overall connectivity map. And once again, hepatic fibrosis and neuroinflammation signaling pathway were the most significant canonical pathways involved. But it's also important to note that molecular mechanisms of cancer had a lower p-value of significance in this overall pathway, meaning that cancer has a greater role, or the molecular mechanisms of cancer had a greater role in this pathway. And with the upstream regulator analysis, once again, the cytokines are up there as the most significant upstream regulators, but this time they have higher p-values, while IL-6 has that lower p-value. So to quantify the data that I found, I used the analysis laid out by Kramer's paper that measures the effects of causal molecules on their target molecules. The distribution of changes of these molecules can be normalized because of their randomized relationships. And a local z-score can be generated from this. And this algorithm was used to confirm a confidence level for that expression analysis. So these are the individual molecules. Each dot represents an individual molecule. And the value of the z-score represents the degree of activation, positive, or inhibition, negative, of the intermediary molecule on its node, either STAT3 or thymoma. So the overall z-score of these intermediary molecules was 10.64, which corresponds to a p-value of 1.03 times 10 to the negative 25. This means that there is a less than 1.05 times 10 to the negative 27% chance that similar results would be found due to chance. And the positive overall z-score also indicates that net activation in the pathway or the trend towards thymoma onset. So to further validate my findings, I went to the NCBI Gene Expression Omnibus and I obtained this data set, GSC 57892. This data set was collected from human patients diagnosed with either thymoma or TET, and it contained 25 samples from 2014. Using the analysis datasets and list tool, I overlaid the data from this data set on my connectivity map and found, once again, that ethanol exposure would lead to STAT3 expression and thymoma onset. Furthermore, in a preliminary study conducted by INIP, there was a binge ethanol spleen atrophy study of 47 F344 rats. 18 of these rats were given a water control, while 29 rats were given an ethanol treatment. And spleen atrophy was measured in the rats given ethanol, as well as thymic atrophy. And according to literature, ethanol leads to the apoptosis of thymocytes, but not epithelial cells. So taking this point, I laid out the question, what are the mechanisms underlying possible ethanol augmentation of thymic conditions? This is the data from that study, and as you can see, for female rats treated with ethanol, there is a 28% decrease average size in the thymus, with a 22% decrease in male ethanol rats. So to recap, through the elevated expression of STAT3, ethanol leads to activation of thymoma. And public data sets and lab collected data complements this in silico IPA study. The molecular mechanisms of cancer canonical pathway has a significant involvement in the mechanisms surrounding the ethanol augmentation of thymoma.
So my next questions are, how specific is the involvement of this pathway in the augmentation of thymoma? And if we are exploring cancer, how do the molecules associated with MMC solely impact progression of thymoma? I would like to acknowledge Dr. Chang for her mentorship during this process, Dr. Liu for his advice, INIP for their support, Petersheim for allowing me to present my research, and Drs. Urso, Snyder, and Gerlifman for allowing me for judging my presentation. Thank you.
Thank you. The next presentation will be done by Ryan King. The title is Molecules Involved in Overall Augmentation of Pancreas and Diabetes. And then we'll go on.
Hello, thank you to Dr. Chang for the introduction. So I'll start off with a brief, simple anatomy of the pancreas. So the pancreas has the islet cells, the beta cells, so the Langerhans cells, including the acinary cells, ductal epithelium, and pancreatic islet cells that are all at risk due to alcohol use. So pancreatitis, or the inflammation of pancreas, is a potential risk factor for the development of diabetes type 2. There are annually about 250,000 hospital cases, and this has been increasing 20% in over the last decade. And alcohol consumption can also trigger inflammation in various organs, varying from the liver, including the pancreas. And alcohol consumption can lead to the development of protein plugs, which can result in progressive inflammation, fibrosis, and loss of pancreatic cells. So based on this background, there is this loss in glucose metabolism, and this results in our hypothesis that alcohol consumption can enhance the development of inflammation of pancreas and non-insulin dependent diabetes mellitus. I've shortened them to IP and DM2 respectively. So what we have used is both the integration of both in vivo and in silico approaches to elucidate the pathways and molecular mechanisms underlying ethanol-induced IP and DM2. So this is just a short flow chart on the network meta-analysis that we used. On the right, you can see that, on the left, you can see that in the in vivo gene expression profiling of acute pancreatitis, we were basically able to analyze a GSD data set on 119 patients, sample genomes with either pancreatitis or they were healthy controls. We then identified for DEGs through RNA sequencing, and then we uploaded those data to IPA to do further analysis on canonical pathways enriched by these DEGs, which allowed us to conduct the meta-analysis. So this is more in specifics on how we retrieved the DEGs from this CLC Genomics Workbench. So what we did was we accessed the GSC data set with the code here. There were 119 patients, either with pancreatitis or as a healthy control, and they were RNA sequenced using the CLC22 Genomics Workbench to find any DEGs between the two sets. And then they were uploaded to IPA to identify any canonical pathways or upstream regulators that are associated with the DEGs and how they are influenced by ethanol. So once again, the sample size was 119 patients. 87 had acute pancreatitis and 32 were healthy controls. They were analyzed with RNA sequencing to determine that there were about 36,700 DEGs identified from the sequencing. From that 37,700, about half of the DEGs were changed by a p-value less than 0.05. So that's what we kept for further study. And they were uploaded to IPA to compare with how many, with the molecules associated with ethanol to determine how ethanol's effects can be moderated into the DEGs. So there are a total of 441 that overlapped. And in terms of ethanol, the 786 molecules came from the QIAGEN knowledge base that is a part of IPA. It curates several publications that allow you to determine any molecular interactions and then go from there. So there were about 790 molecules associated with ethanol, and then 18,000 that were uploaded from the RNA sequencing as the DEGs, and a total of 441 were overlapping, and then they were used further for a core analysis. So the core analysis allowed us to determine the biological relationships between these molecules and any canonical pathways, and a total of 340 were identified with a p-value less than 0.05. From there, this is a very busy diagram, but I will try and explain it as best as I can. So the 340 canonical pathways were then put into a new pathway, and ethanol was added in order to see how ethanol's simulated exposure can mediate an expression of these pathways. And due to an expressed or simulated exposure of ethanol, which is denoted in red using the molecular activity tool, A total of 57 pathways were downregulated, but 156 were upregulated. This showed that there's a very high, they are very related to each other in terms of ethanol, and this varying degrees of pathways that are being affected by ethanol allows us to determine that there is a certain relation going on. And we moved on further with our study to determine the particular molecular interactions that are occurring to allow ethanol's effects on the DEGs. So moving on to the IPA study, so that was our in vivo. We wanted to identify molecules associated each individually with ethanol, IP, and DM2, and this would have allowed us to determine how these molecular interactions can be affected between these nodes as well as with ethanol. So for instance, what we first did was we overlapped ethanol with IP. We found that there were 56 molecules overlapping between them, and the figure on the right shows the network connectivity. It'll be shown on the next slide as well when I've used the molecular activity predictor tool to simulate the exposure to ethanol denoted in red. And this led to the upregulation or augmentation of the inflammation of pancreas denoted in orange. And the pathway in between shows the molecular interactions that has resulted in the augmentation of inflammation of pancreas. So some notable molecules that have been included in this graph have been calculated using the activity analysis, which is a Kramer analysis, which allowed us to identify a cumulative Z-score of 2.01, which corresponded to a p-value of 0.04 in terms of ethanol's contribution to pancreatic inflammation through these molecules. And on this side, you would show any molecules that were inhibited to result in the augmentation of IP, and then the right shows molecules that were activated to result in upregulation of IP. Similarly, we also overlapped the molecules that were associated between ethanol and DM2. Since we already knew that IP could be upregulated from ethanol, we also wanted to see how ethanol can be affected by DM2 as well. And it was found that a total of 136 molecules overlapped between them. And this is also showing the pathway between ethanol and DM2. And certain molecules have been enlarged in this figure as they have been affected by ethanol and directly affecting DM2 as a result as well. So seen here, ethanol was once again simulated in its exposure using the molecular activity predictor tool that allowed us to determine that DM2 is also upregulated through the intermediary actions of vitamin E testosterone, TP53P10, BAX testosterone, D-glucose, and MIR8. So the reason why these molecules were included instead of all was that these 7 molecules were the only molecules that were having a direct effect, not only from ethanol, but also directly to DM2. Whereas for other molecules, they were either only affected by ethanol or only affecting DM2. So these molecules did show us that there was a direct effect between ethanol directly to DM2. And when these molecules were used for quantitative activity analysis, the Z-score was calculated to be 2.4, and the findings are significant with a p-value of 0.16 in terms of ethanol's contribution to DM2. Lastly, we then overlapped molecules with IP and DM2. This allowed us to determine if there's any effects between IP to cause a DM2 reaction. And so we discovered that there were 76 molecules overlapping between them. This is just showing the pathway once again. And this is also a very busy figure, but I will explain it. The ethanol was added to this pathway as a way to determine how ethanol's effects can be mediated between IP and DM2. And I've enlarged certain molecules that have an effect on both IP and DM2. Most notably, there is MIF. This is an inflammatory molecule that is leading to the activation of DM2. I would like to look further into this molecule as it also is also affecting inflammation of pancreas as well. So in the future, I would like to study how MIF can cause any intermediary effects from IP to result in DM2 as a result of ethanol exposure. There was also the inhibition of insulin and insulin receptors 1 and 2. This can also result in the augmentation of DM2 as a result of ethanol exposure. And the inflammation of pancreas, as you can see from here, there were several cytokines that were resulting in the inflammation of pancreas, most notably include TNF, IL-2, or CD40LG. So in conclusion, there were empirical evidences from the previous figures and slides show that the influence of ethanol on IP and DM2 are significant. The integration of both in vivo and in silico approaches were able to elucidate this, the molecular mechanisms that were leading ethanol-induced pancreatic inflammation in DM2, and the findings between both of them were consistent. So based on the figures seen in the previous slide, ethanol does seem to appear to augment IP by activating cytokines such as IL-2, TNF, and CD40LG. as well as by inhibiting insulin and insulin receptors 1 and 2. Ethanol is additionally able to is suggested to augment DM2 by the expression of MIF, as well as by decreasing the production of insulin. I'd like to thank INIP as well as Dr. Chang and Mohammed Bashir for their help. I'd also thank Dr. Ursel, Dr. Snyder, Dr. Gerlichman for acting as judges. Thank you.
So the next presentation is by Rohan Patel. The title will be Involvement of C-reactive protein in the augmentation of patient from SARS-CoV-2 infection.
Thank you, Dr. Chang, for the introduction, and good evening, everyone. Before I carry on with my experiment, I want to give a little background. So what is the SARS coronavirus 2? Well, it's a virus responsible for the COVID-19 pathology. In fact, there have been more than 600 million cases of the COVID-19 infection, with over 6 million deaths related to it. Patients infected with the SARS coronavirus 2 develop respiratory illnesses, and they are marked by acute respiratory distress. However, they also induce a cytokine storm, shown in this figure right there, which can possibly overactivate or inhibit certain molecules. This in turn can affect other organ systems, which can lead to death potentially. So how is COVID-19 related to depression? Well, there have been multiple studies showing the correlation between COVID-19 and multiple neurological conditions. In fact, studies show development of depressive symptoms after the original COVID-19 infection in patients treated as inpatients. The figure on the right is taken from Paquette et al.'s paper, and in 2021, they did a hospital study on inpatients treated from the COVID-19 infection. They list the symptoms that these patients show, and of note, anxiety and depression are there. They're divided into two time frames, 1 to 90 days and then 90 to 180 days. And of note, anxiety and depression have a high incident percentage. So what is depression? Depression is a psychiatric mood disorder characterized by dejection and extreme sadness, and it can be triggered by environmental stimuli or many biological abnormalities. Neuroinflammation has been shown to be a key factor within the clinical signs of depression. So this can indicate a role that inflammation has on depression. So how do we screen for inflammation? Well, there's a protein marker called C-reactive protein. It can help indicate systemic inflammation within the body. And usually in emergency rooms and hospitals, they have a complete blood count test, which can screen for C-reactive proteins. And this can hint abnormalities within the body. And multiple studies shown the increased circulation of CRP noted in post-COVID-19 patients with depressive symptoms. This may hint the role of inflammation in development of depression. With these premises, we hypothesize that COVID-19 increases the expression of CRP, which modulates the development of depression. To start off, I used Ingenuity pathway analysis, IPA tools. These tools were described by my lab mates Sharman and Ryan. I'll give you a quick rundown. The QIAGEN knowledge base is a database that contains the information taken from previous studies, and it can be implemented within the IPA to conduct further analysis. I used the GROW tool. To grow out molecules associated with COVID-19, found 993 molecules associated. I also used it on depression and found 748 molecules associated with depression. Then I used the compare tool and found 144 overlapping molecules between the two nodes. And of note, I found CRP to be one of the 144 overlapping molecules. This could hint the involvement of CRP in the modulation of depression. It could also hint the role that inflammation has within depression. With this being said, I was able to create a network map from CRP to depression. I used the Path Explorer tool to find 20 molecules affected by CRP that affect the development of depression. And with this pathway map, I was able to use the molecular activity predictor, the MAP tool, to activate CRP and find the potential augmentation of depression shown by the slightly reddish orange tint. Of note, CRP increased three molecules: tumor necrosis factor, TNF, interleukin-1, and interleukin-1 receptor antagonist, IL-1RN. CRP increased tumor necrosis factor and interleukin-1. These two molecules were shown to augment depression. However, CRP increased interleukin-1 receptor antagonist, IL-1R, which in turn decreased or inhibited the development of depression. So there are many contradicting findings, so I was able to do the downstream quantification analysis of CRP to find the total Z-score. The same statistical analysis done by my labmates Sharman and Ryan was used. So I found that CRP augmented the development of depression with a Z-score of 0.4021, which is corresponding to p-value of 0.6876. This is not statistically significant. This is not statistically significant, but it does not suggest COVID-19's involvement in depression. It only suggests that CRP's downstream effect on depression might not be significant. To conclude, to wrap up, clinical studies support a positive correlation of post-COVID-19 infection with depression. However, my study focused strictly on CRP. There are other inflammatory molecules or other molecules which may have been inhibited or like overly expressed. However, in the future, I would like to study those molecules and create a broader, more complete picture. Environmental factors do play a role in the development of depression. However, this study focused strictly on the biological mechanisms. Environmental factors such as the isolation from the quarantine, death of loved ones, and the consequences of the quarantine are not factored in. Third, COVID-19 is a relatively new disease, which can limit the amount of circulating information. Therefore, it was hard to access in vivo and in vitro data. Finally, the identification of molecules and pathways through IPA can lead to further in vitro studies. And with that, I want to express my gratitude to Dr. Chang, my mentor, for her guidance. I'd also like to thank Dr. Chen and Dr. Chan in Taiwan for their support. I'd like to thank my NIP lab members for their guidance and support, the Petersheim Academic Exposition Planning Committee for organizing this event and giving me the opportunity to present. And finally, I'd like to thank Dr. Chang, Dr. Urzo, and Dr. Snyder for taking the time to judge. Thank you.
Thank you, Roman. Okay, the next presentation will be done Jonathan Zhang. The title is Studying Signaling Pathway Involved in the HIV-mediated addiction using in silico cybernetic knockout of dopamine neurotransmission genes.
Thank you, Dr. Chang, for the introduction. So today I'll be talking about HIV-1. So HIV is human immunodeficiency virus, which is a virus that attacks the body's immune system. And if HIV is not treated, it can lead to AIDS. Symptoms of HIV include fevers, sore throat, swollen lymph nodes, rash, fatigue. There's currently no cure for HIV. However, people who get treated for HIV, the combinational antiviral therapy, do live longer and have healthier lives. So addictive substance abuse and addictive substance use is more prevalent among HIV-infected individuals. In this table, it looks at the percentage of HIV-infected individuals who use different variety of substances, and then compared to general population. And we can see that in each of these percentages, there's more is more prevalent in HIV-infected individuals than the general population. So neuro-HIV and its use of addictive substances. So the use of addictive substances in HIV patients dysregulates the neurotransmitter-dependent pathways, and these pathways are relevant to and important and critical to the neuronal reward circuit. HIV pathology affects the brain, specifically leading to neural inflammation, and neural inflammation causes neuronal damage and dysregulates the dopaminergic system. So previously in our lab, we did a study on HIV transgenic rats, which mimics HIV patients on CART, the combinational antiviral therapy. And we had 24 rats. So we had 12 F344 control rats, and then we compared that with the HIV transgenic rats. And we did an RNA sequencing of the brain samples, specifically the striatum, the prefrontal cortex, and hippocampus regions. And then this data set was uploaded onto the GEO database, which is part of the NIH's public library, as listed as GSC 47474. So the HIV transgenic rats mimic HIV patients through on CART through a knockout of the GAG and POL genes. by knocking out the gag and pull gene, it makes the HIV virus, specifically the HIV rats, non-infectious, but it also allows the production of HIV viral proteins. So knocking these out removes assembly and reverse transcriptase, but keeping the viral protein still made. So this is a graphical summary of the genes enriched in the neural behavior deficits found in HIV. Specifically in the study, I'll be focusing on drug addiction and the neural transmission dopamine neurotransmission factors, specifically upregulation of PPM1L, PP2CA, CAMK2B, as well as the downregulation of PP1, P14A, DRD4, and GNAL. So the objective of this study is to understand these molecular signaling pathways underlying drug addiction in these HIV patients treated with CART, as well as explore the roles of dopamine neurotransmission genes, the genes I mentioned before in HIV, and also show you an innovative use of in silico tools to knock out genes compared to the contemporary method of using CRISPR-Cas9 to knock out genes. So my hypothesis is that these 6 dopamine neurotransmission genes mentioned before are affecting addictive behavior in HIV patients treated with CART. So summary of my study is basically I'll be looking at these genes and I'll be knocking these out, these dopamine neurotransmission genes, in hope that they will decrease and reduce the drug addictive effects. So graphical summary of my workflow. So I took the GSC 4747 dataset from the GEO database, and then I analyzed it using CLC Genomic Workbench, and then I acquired these differentially expressed genes. From there, I performed a knockout, and then I did core analysis of the knockout, and then core analysis without the knockout, using IPA core analysis, which allows me to find the enriched canonical pathways. So how did I get the knockout? So I used an IPA in silico knockout by removing genes and associated molecules from that differentially expressed genes data set. So there are 1,462 molecules associated with dopamine and dopamine neurotransmission. And then of those molecules, 102 overlaps with the 1,515 HIV differentially expressed genes. And then I removed those 102 overlapping molecules to get the knockout data set, which gives me 1,413 molecules to study. So when I put them into core analysis, when I analyzed them using core analysis, I identified enriched pathways, which are pathways that are either specifically activated or specifically inhibited. Orange represents the activation, blue represents inhibition, and I looked at these different data sets. So I looked at HIV transgenic, no knockout, nothing, and then I looked at the group knockout, knocking out all 6 of these genes, and then I looked at each individual knockout. So of those 108 pathways that were shown before, there are 8 pathways that are specifically related to addiction, and these are serotonin signaling pathway, opioid signaling pathway, neuroregulating signaling, ERK5 signaling, E1F2 signaling, dopamine DARK32 feedback and CAMP signaling, Krebs signaling in neurons, and corticotrophin releasing hormone signaling. And I'll be focusing on opioid signaling, dopamine DARK32 feedback and CAMP signaling, and Krebs signaling today. So when I found these signaling pathways, I did individual knockouts of each specific genes, and then that gave me this table on the right, this figure on the right. So analyzing each pathway, I'll first talk about the Krebs signaling pathway. So psychostimulants, nicotine, and ethanol primarily upregulate the phosphorylation of Krebs. So as we can see in HIV transgenic rat, the black bar, it's significantly upregulated. However, when we introduce the knockout, the red bar, the dopamine neurotransmission knockout of all 6 of these genes, there's a decrease in that activation. And each specifically, the GNAL, the CAMK2B, and then the PP2CA are also decreased compared to the HIV transgenic activation. So when we knock out these dopamine neurotransmissions, it leads to a decreased activation of Krebs signaling, and that could be suggesting decrease in drug addiction. This is because a study done by Krasinova showed that mice, when we deleted Krebs, exhibited decrease in opioid withdrawal behaviors, which suggests that by removing these dopamine neurotransmission genes, it might also have the same effect. So next, I'll be talking about the second pathway I'll be examining in my study, which is dopamine DARP32 feedback and cAMP signaling. So as I mentioned before, the larger the HIV transgenic Z-score of activation is, the more activated the pathway is. And when we produce a knockout, it decreases that activation. And these are found within PP1R14A, cAMK2B, PP2CA, and PPM1L knockouts. So the role of dopamine DARP32 feedback is that it is phosphorylating these addictive substances, alcohol, opioids, nicotine, and psychostimulants are responsible for phosphorylating this DARP32 compound, this dopamine compound. And DARP32 decrease in expression leads to a lack of condition-place aversive behavior in opioid-addicted animals. So when we see this decrease of activation in these knockouts, we can suggest that there's a decrease in drug-addictive behavior and possibly opioid-addictive behavior. And the last pathway I'll be looking at is opioid signaling, where when we knocked out the dopamine neurotransmission, all 6 of those genes together, there's a big decrease in activation, which suggests that this opioid signaling is decreased when we remove them, and these molecules are responsible for that drug addictive behavior, specifically opioid signaling. And by removing those molecules, those specific genes cAMK2B and PP2CA, we see there's a decrease in opioid signaling and possibly a decrease in opioid addiction. So these are all the other pathways that I looked at, and they all show that it reduces drug addictive behavior. So in conclusion, using bioinformatics tool is an efficient and fast method for complete knockout compared to the contemporary CRISPR-Cas9 method. And also, I also found that differentially changes in signaling pathways reveal that the role of dopamine neurotransmission plays an important role in regulating signaling pathways relating to addiction. I also showed that the potential knockout of these genes, those specific 6 genes that I mentioned, could reduce the addictive response and addictive behavior in HIV patients on CAR therapy. So for in the future, I'd like to explore the specific mechanisms of these genes and then how these knockouts of these dopamine neurotransmission genes would be affected in vitro and in vivo models. So these are my references. So I'd like to acknowledge and thank Dr. Sulie Chang, the I9P members for their support, as well as the Petersheim Academic Expedition Planning Committee. I also like to thank Dr. Urso, Dr. Snyder, and Dr. Gerkelman. Thank you for listening to my presentation.
Please, gather the letter up for questions.
Mr. Chang, first of all, amazing presentation. Very well. And that was some original research I yet to see, so I had no idea you could actually do in silico knockouts. So that was very impressive. I just wanted to ask you, so has there been any research done on whether or not the in silico method of knockout is confirmed by in vitro studies or in vivo studies? Is it possible to identify whether or not IPA can be confirmed in an experimental setting?
So basically how IPA works is how it takes the QIAGEN Ingenuity pathway, QIAGEN Ingenuity knowledge base, which is a knowledge base of over 12 million literature findings. And these literature findings are backed up by like in vivo research and in vitro research, as well as in silico research. And that all together backs up this database, which generates this predictive algorithm of seeing how the canonical pathways are involved.
So the predictive algorithm is based on machine learning essentially, and this would be a predicted pathway. It would still need to be confirmed in an experimental setting, correct?
Yes. Thank you for your question.
Yeah, you did a great job.
Yeah.
I have a question for Charmin. So some of the upstream regulators of STAT3 that you identified are pro-inflammatory cytokines that can activate other signaling pathways involved in tumorigenesis. Did you come across anything in IPA or in the literature about other signaling pathways that might be working with activated and deactivated?
So looking at a synthesis of different canonical pathways, I would have to manually go ahead and do that with IPA. With the study, I was more so focused on the individual molecules themselves. So I wasn't really looking at the canonical pathways. And since my study was concerned with thymoma, a cancer, I want to explore more so the cancer side than the ethanol side, because the ethanol, like, there's a lot of literature behind how it affects inflammation, but I want to look at how it affects cancer, and in this regard, like this.
Also, I noticed on one of your earlier slides, it says that this has a low rate of metastasis.
Yeah.
It remains encapsulated. So STAT3, activated STAT3 has been shown to play roles in tumorigenesis as well as metastasis. Is that something also you're going to look into?
Yeah, I think that's a really good thing to look into. Yeah.
Thanks.
Any more questions?
Yeah, I would like to ask Mr. King a question. So out of all the molecules that you found, obviously it's difficult to narrow it down. There's a whole bunch. But out of all those molecules, which do you think were the most important?
I would say in terms of insulin and receptors, I noticed that due to ethanol being simulated and exposure, it resulted in both DM2 and IP augmentation. If insulin is causing both to occur, this would lead me to assume that there's more to this. I hope that I can in the future look into how insulin downregulation can also affect glucose levels, and how this can affect the beta islets and alpha islets, how they interact with each other to result in DM2. Because in this study, I know I just mentioned like simple molecules that are resulting in like the activation or augmentation. In the future, I hope that I can look into how these molecules are interacting with additional molecules or other canonical pathways that can result in DM2. But yeah.
Any more questions? Thank you all. Okay, so the next presentation will be done by Mohammad Bishal. The title is Gender-Dependent Integration of Opioid-Dependent Genes and Signal Pathway in the Strain of HIV Transgenic Rats at the Biomedical.
Thank you, Dr. Chang, for the kind introduction. Good afternoon, all. So the HIV virus is a lung virus which infects the host by binding to the chemokine receptors on the CD4 cell. And seeing the global perspective of HIV, there are 38.4 million people living with HIV in 2021, and every year there are 1.5 million new cases of HIV. So the combination of antiretroviral therapy is a combination of antiretroviral drug which targets at the various stages of the life cycle. The CAR therapy has been very successful in controlling or preventing the infection. However, the neurological abnormalities that has been induced by the HIV virus has not been addressed by the CAR treatment. So the HIV transgenic rat model is a transgenic rat model which was treated by deleting two genes, that is the GAG and POL gene in the viral genome, which is essential for the viral assembly and replication. So this transgenic rat model mimics the HIV patients on CAR treatment. So the misuse of opioid substances, particularly morphine, is one of the major comorbidity in HIV patients. And reports, a previous report from our lab has shown that there is an increased opioid consumption, addiction, and dependence in these HIV patients compared to the general population. And there is a heat map showing the percentage of people living in USA with HIV patients attributed to injectable opioid drugs. With this premise, the present study aimed to understand the modulation of opioid dependence genes and the signaling pathways in HIV patients. We hypothesized that the increased dopamine released to the ventral striatum, opioid addiction, and dependence seen in HIV patients is due to the interplay of overlapping signaling pathways. Coming to the experimental design, we took both male and female F344 and HIV transgenic rats, and these rats received either morphine or placebo pellets. On day one, they received two pellets, and day two they were given 4 morphine pellets by the subcutaneous pelleting. And on day 5 at morphine tolerance, they were sacrificed to take out and the brains were collected, and the prefrontal cortex and the striatum were isolated. Prefrontal cortex and the striatum were isolated, and the samples were prepared for the PCR array of the 65 opioid dependence genes that were previously identified from the QIAGEN knowledge base. In this today's study, I will be presenting on the data from the striatum. So when we studied the expression changes in these 65 opioid dependence genes, and we found when we performed the statistical analysis, we found that some of our fold change values were above the level of significance, that was the 0.05. However, we don't want to drop these the fold change values of these genes because by considering the fact that our sample size was lower, which was n equal to 3, thus we took the bioinformatic approach to using the string and Ingenuity pathway analysis, where we uploaded the fold change values of these 65 opioid dependence genes along with their p-values to the IPA to understand the signaling pathways associated with these opioid dependence genes based on their fold change and p-values. So first we constructed a interaction network using the string analysis, where it gives the clusters of closely related genes among these 65 opioid dependence genes, where we had the calcium channels, opioid receptors, dopamine receptors, glutaminergic receptors, NMDA receptors, adrenal receptors, and cholinergic receptors. Looking to the expression changes in these genes, we found that in males, there were 12 genes were found significantly upregulated in F344 rats, which were highlighted in blue, and 15 genes were found to be significantly upregulated in HIV transgenic rats. In females, there were 4 genes upregulated in female F344, and 1 gene was found to be upregulated in HIV transgenic rat. So we did the core analysis of these 65 opioid dependent genes, and we found that in both male and female HIV transgenic rats, we found the CRUB signaling in neurons and the neuroinflammation signaling pathway was the top enriched canonical pathway that has been associated with the 65 opioid dependent genes, and this was highly regulated with the upstream regulators, that was interleukin 6 in case of male, whereas GCGR in females. We also found that certain functions like conditioning, phase preference, analgesia, and shock functions were enriched in males, whereas conditioning was found to be enriched in female HIV transgenic rats associated morphine. So then we compared this the p-value of overlap between of the signaling pathways among the different treatment groups, and we found that in the HIV transgenic rats received placebo treatment, we found that there were 130 signaling pathways that were totally upregulated in placebo-treated groups. And from there, we found that 26 signaling pathways could potentially contribute to the opioid dependence and addiction in these rats. And then we compared the gender-dependent changes, and we found that there was no gender-dependent changes in the enrichment of these signaling pathways. And the next our goal was to identify the overlapping signaling pathways in the HIV transgenic rats received morphine, and we found that to find that we took the F344 morphine-related signaling pathways and the placebo-treated trans pathway, and we found that among them, 98 signaling pathways were found to be enriched in the HIV transgenic rats received morphine. So among them, these 26 signaling pathways, which were previously found to be associated with the opioid dependence, were also found. Similarly, in females, we found 81 pathways, and the 26 signaling pathways were also found to be enriched in female HIV transgenic rats. We also performed a gender dependence comparison study of these signaling pathways, and we found that among the 26 signaling pathways, 22 signaling pathways were highly enriched in male HIV transgenic rats received morphine compared to the female HIV transgenic rats received morphine. Our core analysis emphasized 2 signaling pathways. One was the CRUB signaling pathway, and the other was the neuroinflammation signaling pathway, which can be potential contributors towards the increased morphine addiction and dependence in these HIV transgenic rats. Coming to the conclusion, in this study, we integrated both in vivo and in silico approaches to identify the signaling pathways that has been associated with both in both male and female HIV transgenic transcripted morphine, and we found that the CRUB signaling in neurons and the neuroinflammation signaling pathway could be the potential contributors. We also identified the 26 signaling pathways and also found the gender-dependent changes where male was found to be more prominent in morphine had more prominent action on the male HIV transgenic tracks. I would like to acknowledge Dr. Chang, Wenfei Huang, and all my NIP members, Dr. Sarir, who is a collaborator in this project, NIH and the Petersheim committee for giving me this opportunity, Dr. Russo, Dr. Snyder, and Dr. Gaiman for judging this. Thank you.
Thank you, Michelle. The next presentation will be done by Wenfei Huang. The title is Molecular Mechanisms Underlying Modulatory Effect of Alcohol Use on Pain.
Thank you, Dr. Chen. I will present on alcohol and pain today. So first, I want to start with a little bit background about the relationship of alcohol and pain. So it's known that alcohol and pain has a complicated relationship. Acute alcohol at like one drink for women or 2 drinks for men in one day can have the pain reducing effects by reducing the pain threshold. Excessive alcohol use can result in tolerance to the alcohol's antinociceptive effects. And further chronic use and further withdrawal from the chronic use can increase the pain by increasing the pain sensitivity. And there are various mechanisms proposed how this happens. Such as the estrogen receptor mediated rate regulation, but it's still unclear. So pain is defined as unpleasant sensory and emotional experience that associated or reassembling that associated with actual and potential tissue damage as defined by the International Association for the Study of Pain. And it can be induced by either the nociceptive stimuli, which is caused by the actual tissue damage, or the neuropathic stimuli, which is caused by the nerve system lesion. And alcohol-induced pain can be from either stimuli. And we have a previous in vivo study using B6 mice that's tested using a hot plate test, analgesia test. The mice were given 3-day binge ethanol treatment, and they were tested in a hot plate test after giving a cumulative dose of morphine. And as you can see here, the latency in hot plate test and the GCA test was shifted to the left. That means the ethanol enhanced the analgesia. So with the powerful tool of Ingenuity pathway analysis, as all my group members has introduced, I'm going to explore the molecular mechanisms that how alcohol use can impact on pain. First, the pain regulation networks will be identified by mining the QIAGEN knowledge base, using the co-analysis of the pain-related molecules, and also by searching the library of the signaling pathways. And then ethanol's effects will be studied by using the gene expression datasets that's retrieved from the GEO, which is Gene Expression Omnibus. And by applying this ethanol-induced gene expression to the identified pain regulation networks, we can identify the possible effects from ethanol on the pain perceptions. So first, the PEM-related molecules was identified based on mining of the QIAGEN knowledge base. And there are 1,958 molecules associated with PEM identified that include 919 chemicals and drugs and 1,039 genes, groups, complexes, and microRNAs, which is shown in the pie on the left. And the co-analysis of these genes, groups, complexes, and the microRNAs reveal the pathways that these molecules are heavily involved. So on the right, it shows the top 10 neurotransmitter and nerve system signaling pathways that molecules are involved. And as another way to identify the pain regulation molecule pathways, the IPA library was searched using the keywords of pain and its synonymous and antonyms, including nociception, analgesia, analgesia, and ache. And 3 pathways were identified. And here, which are neuropathic pain signaling in dorsal horn neurons, opiate signaling pathway, and oxytocin in spinal cord signaling. And all in three pathways, pain were affected through different signaling cascade. And here, the related cascades were highlighted. And then I retrieved the gene expression data sets to study the effects from ethanol treatment from the open public available Gene Expression Omnibus database. The first database I got is the GSE 15774, which is from the striatum of male B6 mice that given one dose of ethanol at 2 grams per kilogram, which is a binge level. And the second data set I got is the GSE 13524, which is from alcohol preferring rats that given 8 weeks continuous ethanol treatment. And by co-analysis of this gene expression data sets from the alcohol-treated rodents, we can see like a differential effects on the pathways were identified. Here I highlighted 2 pathways that were identified to be very significant in pain regulation. And the single dose ethanol inhibit the opiate signaling, but this is not inhibited in the 8-week chronic ethanol treatment data sets. And in contrast, the neuropathic pain signaling in dorsal horn neurons was inhibited by the chronic alcohol exposure, but was not affected by the single dose. And then I focused on each of the pathways that's identified that regulates pain. So here I can see a clear contrast by the single dose versus chronic alcohol use change. So the single alcohol use can induce the elevation of ELK1, prior inhibition of force, and CRAB1, which can lead to the inhibition of neuropathic pain. And in contrast, the 8-week chronic ethanol that can induce
No, it was on silent.
So that will be increase the elevation of force, CRAB1, and induce the neuropathic pain. And the second pathway I'm looking at is the opiate signaling. And similarly, the single dose ethanol can induce the inhibition of analgesic tolerance, which is another pain-related note. And by decreasing the analgesic tolerance, the single dose ethanol can decrease pain. And in contrast, the 8-week chronic ethanol can increase that analgesic tolerance, which can increase pain. And the third pathway, the oxytocin spinal neural signaling, says the pathway was not significantly affected by both single dose ethanol and chronic ethanol dose. Here we can see that the analgesia node was not affected here. And also the upstream analysis of the 2 data sets both revealed the ESR1, which is the estrogen receptor 1 as the top regulator of this observed data gene expression differences, changes. So we can say that the first one, the CRAB1 and the FAST1, which was identified in the 2 pain regulation pathways that were regulated by the estrogen receptor 1. So and also estrogen receptor is, there are evidence that estrogen is capable of modulating of the activity of those sensory neurons in the nervous systems. However, the mechanism is unknown, but here shows the proposed mechanism by other group, like they can impact several systems, including the opiate systems that regulate the pain. So to further confirm if estrogen receptor 1 can play a role in the ethanol's differential effect, I do a so-called estrogen receptor 1 knockout that Jonathan has just introduced. Like we remove the estrogen-related molecules from the data set and see how the pathways will be affected. And here we can see that by removing the estrogen receptor 1 and its related molecules, the 8-week chronic ethanol's effect on the neuropathic pain and the analgesic tolerance was removed. By the estrogen receptor one knockout in the single dose ethanol data set, it doesn't affect that much. So as a summary, we have seen that single dose or short dose ethanol exposure can have anti-nociception effects, and chronic ethanol exposure can lead to pain, which can be regulated through the neuropathic pain signaling and opioid signaling, and very likely through the estrogen receptor 1 and its regulated signaling pathways. And I would like to thank Dr. Sulie as my mentor, and thank my lab members, especially Bishur, and I thank Dr. Chang from the NCCU, and thanks to Petersheim Expo for giving me the opportunity to present, and thanks Dr. Russo, Dr. Sandra, and Dr. Denman for taking time to join. Thank you.
Thank you. The next presentation will be done by Tatiana Richivo. The title is RNA sequencing identifies variants in NAVA areas for cleft and palate. Thank you.
All right, thank you, Dr. Chang, for that kind introduction. So let's get started. So a little bit of background. So cleft lip and cleft palate are birth defects characterized by a split or opening in the upper roof of the mouth in the case of cleft palate, or in the upper lip as the case of cleft lip. And these defects form while the lip and the mouth of the fetus start to develop between the 4th and 9th weeks of pregnancy. So according to the CDC in the United States, 1 in 1,600 babies have cleft lip and palate. 1 in 1,700 babies have cleft palate, and 1 in 1,800 babies have cleft lip without cleft palate. So in terms of complications of cleft lip and cleft palate, these include difficulty eating, like increased likelihood of ear infections, difficulties in communicating, and dental problems. So surgery is the most common form of treatment. So for cleft lip, it can be done anywhere between 6 to 10 months at the earliest, and then 10 to 12 months for the palate. So in terms of genetic causes, there are no known ones as of yet. So hence why we decided to do this study. We utilized bioinformatics software, specifically Ingenuity pathway analysis and CLC genomic workbench, in order to analyze RNA sequencing data. And this RNA-seq data was collected from the Sequence Read Archive, which is curated by NIH. And we have the ID that we specifically used from it. So in this sample, there were 6 whole blood samples from patients ranging from the ages of 3 to 6 months. And 4 of these were cleft lip and palate patients, and 2 of them acted as controls. So in terms of our study goals, we wanted to look at transcriptomic expression in cleft lip and palate patients versus the healthy controls, gene enrichment analysis in relevant pathways, and single nucleotide polymorphisms, or SNPs for short. So going into transcriptomic expression in these patients, we put the RNA-seq data into CLC genomic workbench, and through this we were able to find the false discovery rate, p-values, and the absolute fold change for the differentially expressed genes, or DEGs for short. And as you can see here, these were the genes that were found. So we were able to plot these in a volcano plot. And as you can see here, the blue represents downregulation and the red represents upregulation. So going into our gene enrichment analysis, we put our DEGs into IPA and we ran a canonical pathway analysis. So what we were able to get were the physiological system development and functions, as well as the diseases and disorders. And some of the more significant ones, as you can see here, are embryonic development, inherent skin development and function, as well as organ development. And for diseases and disorders, we have cancer and organismal injury and abnormalities. So looking a little more into it, we put the rest of the data that we had into ShinyGo. This basically is a software that's able to graphically represent our data. So as you can see here, looking at our biological processes, IPA was able to see some of the More significantly enriched ones were cornification, skin development, keratinite differentiation, epidermis development, and epidermal cell differentiation. So looking into our single nucleotide polymorphisms, 47 SNPs were found, and they were containing 20 additional genes. And of the observed SNPs, 10 were predicted as possibly damaging, and 15 were predicted as unknown. So for MUC7, there was one SNP detected, as you can see here, and this SNP was predicted as unknown. So the reason I mentioned MUC7 was because due to its significant downregulation observed in CLC, we wanted to observe the genes that were associated with it. So MUC7 encodes for mucin7, which is a small salivatory mucin found in the minor salivatory glands that act as a protectant against bacteria. So as you can see in this chart made in IPA, we were able to find several genes that were associated with MUC7, and several of them were related to the mouth in some capacity.
And two examples I wanted to highlight here. As you can see in the extracellular space, we have AMY1C. So this is actually a gene that encodes for an enzyme that helps in protecting against bacteria in the mouth. And as you can see in the cytoplasm, we have FLG. While this isn't restricted to the mouth necessarily, it does assist in epidermis development. So in terms of our conclusions, MUC7 was found to be significantly downregulated in cleft lip and cleft palate patients and associated with genes in the oral cavity. And our data provides strong evidence for the role in MUC7 for cleft lip and palate. As you can see in our previous slides, we found that they were associated with genes in the mouth and also with our SNP, and then it was significantly downregulated. And our study has served as a stepping stone in finding the genetic causes of cleft lip and cleft palate, and currently this study is in preparation for publication in the near future. We submitted it to Nature. We're really hoping we get it. So in terms of acknowledgements, I would personally like to thank my mentor, Dr. Sulie Chang, for allowing me to collaborate with her. I would also like to give a shout out to my main collaborator, who unfortunately could not be here today, Dr. Rachel Montel. I would also like to thank Dr. Rastami. He was also a main contributor in this project. I would like to thank the members of the Institute of NeuroImmune Pharmacology for their constant support and guidance. I would like to thank the Seton Hall University Petersheim Expo Planning Committee for allowing me to present this. I would like to thank our 3 judges, as you can see here, for agreeing to judge this. And I would like to thank NIH for the grant that they supplied that helped fund this study. So thank you all for listening.
The next presentation, last one is Jen's follow. The title is Comparative Analysis of the Immunological Clinical Pathway and the Upstream Regulator Differentially Modulated in Response to Conditional Bone Fracture.
Thanks for that introduction, Dr. Chang, and good evening, everyone. I'm happy to go last today. Thank you for your time, attention, and patience. So just a quick background. So the cellular and molecular mechanisms that underpin bone fracture healing are highly regulated and very organized. We have different cell types, molecular factors that are localized in the bone fracture callus in a location and time dependent manner, including the 3 primary bone-related cells: osteoclasts, which resorb the bone matrix; osteoblasts, which produce the hard bone matrix; and chondrocytes, which produce extracellular matrix and cartilage, which is a key intermediary factor in the healing process. This also includes immune cells and the factors they express and secrete, which localize in the fracture callus, also in a time and location dependent manner, and influence factors like recruitment of stem cells to the callus, their differentiation and function into mature bone-related cells, as well as the overall resolution of the fracture into morphologically complete, structurally sound, and functional bone matrix. This type of research is part of an emerging field with respect to the interactions of immune cells, the immune system, and bone that is defined as osteoimmunology. So with complex mechanisms, as I described, many things can go wrong. And when those things go wrong, you experience what is called a non-union or malunion, basically the failure of the bone to rejoin and become structurally sound again. This has a number of different risk factors associated with it, including genetic and epigenetic risk factors, lifestyle factors such as alcohol consumption, obesity, smoking, pathological risk factors, diabetes mellitus being one I'm interested in my dissertation research, as well as certain medications that all can contribute to the risk of fracture malunion. In context of diabetes, it is understood that a combination of the metabolic syndrome, cellular and immune dysregulation contribute to the risk of malunion, but the exact mechanisms that occur in this context are not well understood. So in this context, I hope to use IPA as well as publicly available data sets evaluating bone fracture in some condition to identify and establish pathways that are universally dysregulated or modulated in the fracture healing process, in hopes of curating a list of genes and pathways that I could use to prioritize my dissertation research. So in terms of the methodology and design, I wanted to establish a comparison across multiple data sets. This is challenging because those data sets are generated in different means through different platforms with different conditions and reference standards. So I first sought to identify a controlled data set that reflects normal fracture healing processes with respect to changes in gene expression. From there, I identified several data sets within which fracture healing was perturbed through some experimental condition. So in effect, I'm comparing normal changes to disrupted changes. In that context, after identifying the relevant data sets, I confirmed that the files were actually compatible with CLC and IPA, which is a question you do need to ask. From there, I prepared the data through sequence alignment and performed either RNA analysis or array analysis with respect to the different types of data. The resulting differential gene expression values were then used to perform core analyses across 7 individual data sets, targeting between 5,000 and 7,000 genes with a nominal p-value no greater than 0.5. From here, I combined and consolidated the different data sets into a comparison analysis focused on canonical pathways and upstream regulators. I then extracted the z-scores and performed a weighted qualitative assessment intending to rank the pathways and regulators that were most different across each pathway and molecule to prioritize them for my research. So on the left, you can see the output from IPA, which reflects each of the data sets, and in this example, the canonical pathways in rows. This is 162 canonical pathways of the 4,500. This is software identified. I did do upstream filtering of the pathways and molecules to focus on immunological factors, which you see here, but still this list is too large to be meaningful, digestible, etc. I've translated this to a table where I've considered the relative z-scores, again, compared to the control data set, multiplied that out through each conditional fracture experimental data set to define a weighted cumulative difference, and then sequence and order the data set by pathways that have the highest cumulative weighted difference. So reflecting pathways within which there is the most difference across data sets relative to the control. And what you can begin to see is that there does appear to be some meaningful biology emerging here with respect to immunological changes that occur in the fracture. Namely, changes in chemotactic signals, the ability of leukocytes to migrate to the fracture and or the stromal cells at the fracture site to produce chemotactic factors that cause leukocytes to migrate, increase in pro-inflammatory signaling through IL-6, IL-33, S100 peptides and toll-like receptors, as well as increases in leukocyte co-stimulatory signaling, namely through CD28, CD40, and 4-1BB, as well as a global general decrease in anti-inflammatory signals mediated by IL-10. Applying the same approach to the upstream regulators, where there was generally less significance or difference in the data sets across each conditional fracture group. Nonetheless, some of the same features that I described before with respect to the canonical pathways are visible here in this context. So we see an increase in pro-inflammatory signals, namely driven by macrophage and granulocyte colony-stimulating factors, the activation of interferons, as well as corticotropin-releasing hormone, which when secreted by T cells is potently inflammatory. Interestingly here, we are seeing increases in signals associated with B cell survival and differentiation, CD40 ligand, TNFSS1-3B, otherwise known as BAFF or GLYCE, as well as hematopoietic modulation, namely through IL-3, as well as, again, indication of decreased inflammatory regulation mediated by IL-10. With all of these factors and pathways, I wanted to assess how they interacted with each other. So I took all the pathways that were common between the data sets as well as the molecules and added them together using the pathway building tools in IPA, and subsequently used the molecule activity predictor to selectively activate targeted molecules. So in the two examples on the left and in the middle, I activated IL-17 alpha, which is a known cytokine that influences the differentiation and function of osteoblasts and osteoclasts. And we see here when this molecule is activated, we see a general increase in the inflammatory state, but also an increase in bone formation, osteoblastogenesis, and osteoclastogenesis, as we would expect based on the literature. Also, when activating CRH, we see that same overall profile and trend, although CRH is not really well described in the literature in context of its relationship to fracture healing. Most interestingly, when we activate CD40, a co-stimulatory receptor expressed on T cells, B cells, and antigen-presenting cells, we see the same general context of broad inflammatory activation. However, we also observe inhibition of bone formation, osteoblastogenesis, which is in contrast to the other pro-inflammatory markers that I've identified here, which would otherwise activate those pathways. So in conclusion, through this work, IPA certainly has validity in being usable to help identify and prioritize pathways and molecules to support experimentation, prioritization, and hypothesis development. With respect to my work, the analysis suggests immune cell migration, chemotaxis signaling, hyperinflammatory conditioning, and hematopoietic conditioning may be areas to further prioritize and evaluate as I conduct my research. As I said, I did perform upstream filtering and or downsampling of the pathways and molecules to create a more manageable list. However, I would like to unfilter those data, use all 4,500 pathways, all 12,000 molecules, and overlay that with some level of gene ontology to maybe include more molecules or pathways that may not be directly related to inflammatory responses, but may otherwise regulate them. IPA is certainly a powerful tool, but there are some considerations. Namely, the software is based on predictive algorithms that make assumptions, and those assumptions can be wrong because they misinterpret the underlying data. So I intentionally included the example of CD40, which in the QIAGEN knowledge base is predicted to downregulate bone formation. But when looking at the source for that relationship, I actually found that the researchers highlighted that CD40 was necessary for bone formation, and when activated, it contributed to bone formation. IPA misinterpreted the relationship because of the way the experiment in that paper was designed. So this is not to suggest IPA isn't a useful tool, but it is to highlight that manual curation, manual review of your data as generated through IPA is really important. So with that, I'd like to acknowledge and thank Dr. Chang and my colleagues in our molecular readings class for their support and help with me learning to use and apply CLC and IPA to address these research questions. Also, I want to thank Dr. Cottrell and my lab mates for their support of me as a part-time PhD student in my dissertation research, and to you, the audience and judges, for your attention and consideration of my work. I've listed my references, including links to the datasets in the Gene Expression Omnibus, as well as a few other references that I used in preparation of this presentation. And with that, I think it is actually time for questions.
Let's have 5 minutes of question. Any question?
Yes, in the back.
So I have a question about your comment about IPA and just being aware its possible limitations. So if you have so many genes involved, like for our food 3, we're looking at all of these genes, and there's so many of them. If we see that something is indicated on IPA as a likely gene, do you recommend just like reading through clicking that molecule, reading through most of the sources that IPA lists to confirm whether or not you want to move forward with that?
So I think that's a good idea in general. IPA does have some tools to help you understand the significance or relevance of the relationships it's predicting or assuming. Namely, when you use the activity predictor, the color of the lines indicates whether there's activation or inhibition, or when it's yellow, that there is kind of diverging data present in the system. So in those instances, I would highly suggest you go in and look at the underlying sources and interpret them for yourself. But if you do, for your research example, pick a molecule to focus on, you want to be sure that your time is worth investing in that molecule. So I would just suggest you review all the underlying literature.
You said in your paper that there are no genetic studies done in cleft palate. Do you have any idea what that is, or is there just not been any funding or interest?
I don't think it necessarily is that there haven't been any studies done. I just think it's more that we haven't had any proven genetic causes thus far. So right now, we're trying to find those underlying genetic causes. So I don't think it's necessarily a lack of people researching. I just think it's a lack of actually finding definitive ones.
Right, so you didn't find any evidence for any kind of genetic basis in the literature yet, so not proof, just suggestions, implications.
We have some major implications, but we're trying to look a little bit further because we were actually wanting to use another data set as well to try to further confirm our results, because I think the main issue with what we have now is that our data set was just so small. It was only 6 individuals. So we want to probably put in another data set which actually has 10 individuals additionally, so we can further confirm that.
I had a similar question, actually. My question was, is there any evidence, even if it's anecdotal, that there's a Mendelian pattern to the inheritance of cleft palate?
So thus far from what I've seen, there is some evidence that there is a genetic component to it. We just haven't found the exact ones.
Sorry to bother you,
No, that's okay.
What about toxicological reasons and malformation in the womb? So what is the balance between genetic and environmental causes, if you have any?
So interestingly, in terms of environmental causes, some of the ones that they've associated with it primarily are smoking and alcohol abuse. So there are some environmental factors that can be incorporated with the genetic factors.
Any more questions?
So with alcohol and you said endorphins or pain?
Yeah.
Yep, pain. So what is the future of research with that? Do we have any evidence that getting people out of alcoholism will reduce their need for opiates and then pain? Has there been any studies done?
Well,
Well, there are, I would say like a lot of studies, or that as a, like, just like the, there's not like a clear mechanism how this happens. Like a lot of people study alcohol and the effect, the transgenic effects, but there are like, people are still studying like how this happens. And let's say like there are differential effects as short versus chronic alcohol. So like how molecular-wise, genetic-wise, how that happens is still unknown. Because yeah, like there are already a lot of alcohol studies, so I feel like it's easy and necessary to use already those like published data sets that we can analyze them, and especially with nowadays those powerful bioinformatics tool, and it gives us like more insights on those like expression data alone.
In your gender comparative study that you mentioned between male and female mice, do you know why male mice are more affected by morphine compared to the female mice?
I can say on a molecular based perspective, like when I studied the 65 opioid dependence genes, I found that the 15 genes were upregulated in male mice when compared to females. Also in the pathways, like the CREB signaling pathway was top enriched, where the CREB actually regulates a lot of proteins and molecules that are involved in that drives addiction. For example, the dynorphin, which is a kappa receptor agonist, so which actually regulates the negative effects of morphine. So it would be a reason.
Any more questions? Okay, so give all the presenters a crowd. So I'll say that 2018, 2019, we began this as an OMI project. And I have to say that I'm very pleased to see the advancement we have moved in now is 2023. And then the use of QIAGEN knowledge-based IPA actually is much more advanced than very beginning when we have faced that. So thank you all. And the food actually waited outside the door. So get out and let the people come to sell the food. And stay back.